Good morning, everyone. It is a pleasure to welcome you at this meeting. I'm very happy to see so many participants. I hope we will have good discussions today. I would already in advance thank the experts that will guide us through the documents today for their participation and their time and for traveling here to this meeting. We will have, after each presentation, a moderated discussion. It would be helpful for us if you could raise your hands if you have any questions or remarks during this discussion time. You can ask questions in the chat, but because there is not so many people available to follow that is a bit more complicated. So it would be great if you can just ask your question by raising your hands during the discussion. Another question to you is that please also provide, if you haven't done so already, any comments that you have to the guideline in writing. That will also be helpful for us to review the guideline one last time. And with that, I would like to give the word to Susana Casado, who is the first speaker. She is with the regulatory authority in Spain and is the vice chair of the Novel Therapies and Technologies Working Party and is also the NTWP representative in the operational expert group that wrote this guideline. So Susana will set the scene. Go ahead, Susana.
Good morning, everyone. Can you hear me well? Yeah? Okay, so I thought to present myself, but I think that the presentation was really nice, so I move directly to my presentation. So my objective with this presentation is to share with you the context on the development of this guideline. Okay, so as of course all of you know, phages are a virus that infect bacteria. They have two biological cycles. We are interested in those bacteriophages that follow the lytic cycle because they are able to break bacteria and then to eliminate them. Next slide, please. So although bacteriophages are making a comeback now, the phage therapy has been used during the last 100 years, and we have very interesting publications that have a review on that. Bacteriophages have been used for a high variety of bacteria strains and also for different target species, as you can see in this slide. And I invite you to review the two bibliographic references that I include in this presentation. Next slide, please. There are striking cases that the one that I'm presenting here and described by Green. In this case, the use of bacteriophages was focused on cure the healing capparas, the healings in the capparas of a sea turtle. It was a treatment of around 1 year, so long treatment, but as you can see in A, at the end of the treatment, the capparas was more or less recovered. Next slide, please. So bacteriophages are not only used as phage therapy, that is our matter today, but they are also used for food biocontrol or disinfectant. And yeah, a good review on this can be found in the publication I include below. Next slide, please. Yeah, the regulation of phage therapy is different by the different authorities. And as an example, the FDA classified bacteriophages as GRAS, generally recognized as safe, and this is an exception claim, so they are not submitted to pre-authorization measures, and they have a reduced dossier. You can see those that are already authorized in the publication of Ateburi. And in Europe, there has been already 2 applications to EFSA, because these bacteriophages are intended to be used as feed additives or decontaminating agents. And EFSA is the body that is in charge of these authorizations. And when bacteriophages are used as medicines, then the EMA is the responsible. Next slide, please. Bacteriophages used as a medicinal product, that means that they are used to cure or treat or prevent a disease, are under the regulation 6/2019, and they are also classified as novel therapies, and this implies some particularities that I will explain briefly in the next slides. Next slide, please. One of the particularity is that this kind of product needs to be registered by a centralized marketing authorization procedure. It's mandatory, so the application for marketing authorization should be sent or submitted to EMA. And EMA compromised to issue an opinion in 210 days. And the marketing authorization is valid throughout the European Union. Next slide, please. The dossier that needs to be submitted to EMA should follow the Annex 2 to the Regulation 6/2019. There, the technical data requirements are explained, and the applicants should follow those requirements applicable to biological other than immunological veterinary medicinal products. The full dossier is like usual, with Part 1 with administrative information, Part 2 quality, Part 3 safety, and Part 4 efficacy. And deviation from the requirements of the Annex 2 are possible when justified. Next slide, please. I think we can move to the next slide, because you can find on these slides a copy of what is stated in the regulation, so you can read there. Next slide. Next one. Yeah. So why we are focused on bacteriophages right now? This is because the new regulation 6/2019, include them in their text, and also because as it is stated in this CVMP strategy on antimicrobials, they are considered ATAMs, so alternatives to antimicrobials. So the CVMP is compromise in providing regulation and scientific advice to try to foster the development of this kind of products. So one of the objectives is to give further guidelines on this kind of products and on the data requirements that are needed, and also to prompt the international cooperation. Next slide, please. So that's why the Novel Therapy Working Party was appointed to develop a guideline, and that was our work during the last year. So when we started working on this guideline, we found some regulatory and scientific challenges that we try to resolve with this guideline, and are the ones that you can see in the slide. So one of the challenge is the characterization and selection of phages and bacteria strains used for production. Another challenge that we identified is the risk of the presence of lysogenic phages during production, the expected need of changing the composition of phages of the products. I think this is the more challenging under the regulatory point of view. Also, the potential risks of this product on the normal microbiota, their potential effect on the immune response, and also the potential release of endotoxins when they are using as treatment. The assessment of environmental safety is also difficult to forecast, and also not only for the genetically modified bacteriophages, but also for the natural ones. The correspondence between in vitro and in vivo efficacy quality controls is difficult, and the design of studies, selection of efficacy endpoints, control time of administration. How to monitor the post-marketing efficacy to ensure sustained efficacy is also a challenge, and the study of the resistant mechanisms of the bacteriophages against these phages. Next slide, please. So, as I said, the Novel Therapies Working Party was appointed for developing this guideline. This working party starts their work in May 2022, and we start with this guideline and another on cell therapies that both are almost finished. And the objective of the novel therapy is to provide recommendation to the CVMP on all matters relating to novel therapies and technologies, and the main duties are the development of guidelines, addressing queries, and contribute to training on evaluation of novel therapies. Next slide, please. So this is our working plan for last year. As I say, we developed 2 guidelines, and now we are working on a guideline on nanomedicines. Next slide, please. And of course, we prompt the international collaboration. In fact, in drafting the guideline, representatives from EFSA and FDA were actively involved. And we also share our versions with EDQM and with the inspection working group. Next slide, please. So this was the timelines of the development of this guideline. We are now at the first quarter of 2023. We are close to the guideline adoption, and we are right now during the public consultation of the guideline that will close at the end of this month. So we invite you to review the text and please send us comments, and that's why we are here today. Next slide, please. Yeah, here is a summary of the concept paper, but you can read it in the EMA website. I think we can move to the next slide. And the next one. And the next one. So what is the aim of this guideline? The aim of this guideline is to establish the regulatory and technical and scientific requirements applicable to this kind of products that have the particularity that needs a flexible framework. And that's what we try to do with this guideline, try to flexibilize the requirements to allow the development of this kind of products. We also want to applicants seek early advice on the development of this kind of product because of the biological complexity and nascent nature of this medicinal product. So I think these are the two messages that I really want to share with you. Next slide, please. So for today, and as Jacqueline already said, we expect that you to review the guideline as far as possible and and send us comments during this period of public consultation. And we would like you to focus on the applicability of the guideline to the current and future developments. And with this, I finish my presentation. And I'm not sure if we can open a discussion or you prefer to move to the next talk. Thank you very much for your attention.
Thank you for that, Susanna. So my question to you is, are there any questions at this point? Oh.
Yes, there's one question in the chat. So, yeah, it's about efficacy. So we can also do it later after the efficacy presentation, perhaps.
Okay, thank you. So we will leave the question in the chat for the more appropriate session on efficacy. And I will ask you again, of course you can put questions in the chat, but we would appreciate if you would ask them by raising your hand during the discussion after each session. So I think, Susanna, that everything was clear. Thank you very much for that. And then...
sorry to interrupt,
sorry?
There's one hand raised. Yes, John Iredell, or Iredell.
Hi, thank you. It's John Iredell from Australia. I just wanted to ask briefly, do you envisage that genetic modifications will be encompassed in the same regulatory process, or do you expect that to be handled separately?
Hi, thank you for your question. You mean modification of the bacteriophages or
Yes. Yeah.
You mean derivatives compound? Yeah, modified bacteriophages are part, are under the scope of this guideline. What is not under the scope is the derivatives of these bacteriophages. But modified bacteriophages in principle are included in the guideline and submitted to what it is stated there. Thank you.
Thank you for that. So if there are no further questions at this point, if you think of any questions to Susanna, you can always ask them later, of course. Then we will now proceed to the session on the quality part of the dossier for bacteriophages. And leading us there will be Martin Oleksiewicz. He is a head consultant with the Danish Medicines Agency and a member of the operational expert group that wrote this guideline. And Martin was specifically involved with the part on quality. So he really is our expert there. So Martin, please go ahead.
Thank you very much, Jacqueline. So I'll be presenting the quality documentation which would be expected for veterinary phage products according to this draft guideline. And you obviously see the title page here on the slide. And as you heard in the introduction by Susanna, the new EU regulation makes special provisions for what is termed novel therapies, which includes phage therapies. So there are two main, I guess, provisions. First, to improve accessibility of novel therapies, the quality requirements that would generally be expected for biologicals can be adapted if justified, of course. And second, it's anticipated that these products may need to be updated on a regular basis to overcome resistance or to adjust the product to changing epidemiology in the field. And so it's stated in the new regulation on veterinary medicines that a flexible composition might be expected for these products. So it should be possible to customize the products to fit the situation in the field. So as indicated here on the slide, I'll focus on these innovative aspects as regards the quality documentation and not go so much into obviously the general requirements for biologics, which I'm sure you know. And the next slide, please. So to take the formalia first, I'm employed by the Danish Medicines Agency, but I'm giving this presentation in a personal capacity, having been a member of the team responsible for drafting the guidelines. And I don't have any conflicts of interest to declare here. Next slide, please. So here you have the table of contents of the draft guideline, and I'll be talking about section 4.2, the quality documentation, and section 5 also, a special section about post-marketing changes to products, because that has quality aspects obviously as well. And as regards these post-marketing changes, the guideline focuses on the specific situations where these changes have to be done to overcome development of resistance or to adjust product to changes in epidemiology of pathogens in the field. And you may also please note that there are 3 annexes in the guideline which also provide quality-related information. And the next slide, please. So from a technical perspective, what is a phage medicine? As Susanna informed us, the phages have to be lytic. So the mode of action has to be killing of bacteria by a lytic mechanism. This is directly stated in the new EU regulation for veterinary medicines. These products can contain 1 or more phages. That's termed monophage and multiphage products in the guideline. The phages can be natural, meaning that they can be isolated from natural sources and used without any modification, obviously with purification and all that, but without modification. Or they can be modified to give them better properties, for example, higher potency or broader host range. And this optimization, as was also discussed in the question just before, it can be done by classical microbiological methods, selection methods, or by genetic engineering. So genetically engineered phages are within the scope of the guideline. Phage particles are also increasingly being used as display platforms, for example, in vaccines or other purposes, and this is not in scope of the guideline. Also, the use of integrating phages to change bacterial phenotypes, for example, to increase susceptibility to antibiotics, would not be in scope of the guideline. And phage-derived products, lysins or enzymes or whatever, are also not in scope. Next slide, please. So one of the innovations in the new EU regulation for veterinary medicines is this stress on regulatory flexibility for novel therapies, including phage therapies. So as stated at the top of the slide, generally quality requirements should be proportionate to the risk associated with the intended use of these products. And that's a very general statement that we probably can all agree with, but where can details be found? Which framework is this based on? So I think it's true to say that this is based on basically current quality risk management principles, for which you can find details in ICH guidelines, for example, which are cited here on the slide. We have, to the extent possible, tried to clarify application of quality risk management principles in the guideline. And some examples you can see here in the gray box. So, for example, indication will play a role. So products with a high clinical impact generally will be considered differently than products with lesser clinical impact. Route of administration obviously will play a role. Requirements to topical products would generally be less stringent than requirements to parenteral products. The biological complexity of products would play a role. So complexity might be, for example, reflected in critical quality attributes. So those are quality attributes critical for safety or quality, and products with many such CQAs or complex CQAs would again probably require a higher level of quality documentation than more simple products, and so forth. But to end all this, these examples, this is a nascent field, as Susanna also mentioned. There are no marketed products. There's no regulatory precedent. So the required levels generally of quality documentation can't be pre-specified and need to be evaluated on a case-by-case basis. And we state that, I think, through all the sections of the guideline, and also mentioning in several places that if in doubt, obviously, there are scientific advice procedures available. At the next slide, please. For phage products, such as for any products really, the quality documentation must include a description of the composition. So the simplest type would be a product with a fixed composition that's illustrated at the top. A product where the components are fixed. So regardless, this can be a monophage or a multiphage product, but regardless of whether it's one or the other, for a fixed product, the identity of all phage strains should be listed in the dossier, in the application for the marketing authorization. And so if a fictitious application text is given in a small box at the top there on the right over the slide, so using a multiphage product as an example, one might apply, for example, for a product containing 3 phages, and so this could be phages A, B, and C. And this is, of course, a type of product which resembles most traditional medicines. So on the biologic side, this would be comparable to, let's say, a cocktail of monoclonal antibodies or multivalent vaccines, or on the chemical side, fixed combination medicinal products, such as, for example, combination of beta-lactam antibiotics with an beta-lactamase inhibitor. But phages are obviously quite specific biological agents, so the host ranges can be narrow. So the products may be sensitive to changes in epidemiology in the field. And also it's considered that resistance can develop relatively rapidly. So these products may need to be adapted on an ongoing basis. And it is therefore recognized in the new EU regulation that a flexible composition might be relevant, might be valuable. For products with a flexible composition, again, these may be monophage or multiphage products, but for these products, the following information needs to be given as listed here on the slide. So a listing of all phages which might be included in the product must be given. A range for the composition must be given, so that's the minimum and maximum number of phage strains in the product. And for each phage strain, and also for the product as a whole, a range for the phage level per unit or per dose must be given. And again, you have like a fictitious application text in the box at the bottom right. And so again, using a multiphage product as an example, and again, staying with 3 phages. So here we are applying for a product where these 3 phages might be selected from a list of 8 phages called A through H here in the example. In the gray text at the bottom, also it's indicated that obviously for fixed as well as flexible products, in addition to providing this information on the phage components, the standard information on excipients and so forth and so on also must be provided. And the next slide, please. This flexible composition is a regulatory innovation. And Susanna, you mentioned that we had many challenges when we drafted the guideline, but I think getting our heads, or certainly getting my head around what this flexible composition means was challenging for me at least. So I'll try to give more detail on this here. The boxes at the top are just to remind what this product with a flexible composition is. So we are staying with this fictitious example. And so first, the first thing to mention is that even though this is a flexible product, obviously a justification has to be given for all phage components proposed for the product. this is, I think, a general requirement for fixed composition products, for products containing more than one active ingredient, rather. Such combinations can only be justified if the combination offers advantages over the individual components, obviously. So for this fictitious product, one would have to justify why it's indeed necessary to have 3 phages in the product as opposed to just 1 phage. And one would have to justify, for example, why the listed 8 phages, A through H, are actually relevant, given, for example, the chosen indication. There are more details on this justification, what types of questions should be answered in Annex 1 of the guideline. It should also be stated that for all the listed phages, these 8 listed phages in this case, A through H, they must all obviously be characterized as regards host range, absence of lysogenic activity, potency for the relevant bacterial pathogens, genetic characterization, and so forth. Details for the generic characterization are given in Annex 2 of the guideline. So on the quality side, all phages must obviously be fully characterized, and this is a part of the section that we've tried to coordinate with colleagues at the European Pharmacopoeia, and there'll be more on that a bit later in the meeting here. And then finally, the phages listed in this for this flexible composition A through H, they have to be characterized, but they don't actually need to all have been tested in safety and efficacy studies. And we'll come back to that later also in the presentation, sorry, in the meeting. But what I can say for now is that if some of these listed phages would not be included in safety and efficacy studies, then the existing knowledge should be sufficiently predictive to justify the registration as part of this flexible product composition. So basically, this flexible composition may not carry with it unacceptable risks for quality, safety, efficacy of the final product. And then to state the obvious is that if one wants to register a product with a flexible composition, then obviously master seed stocks of all the listed phages would need to be available in a biobank, for example. So the phages that are registered for the flexible composition obviously have to be physical existing phages. Next slide, please. To sum up then the general quality data requirements for phage products with a flexible composition, as previously mentioned, this is kind of difficult because it's a nascent field, and so it's difficult to pre-specify the actual required level of quality documentation. Flexibility is expected, but what can be said is that the dossier must be sufficient to document that the flexible composition, for example, does not carry with it unacceptable risks, as mentioned, to quality, safety, and efficacy of the final product, to kind of fulfill this overall overriding philosophy that the requirements for novel therapy products should be proportionate to the risks associated with their intended use. If these conditions are fulfilled in the dossier, then manufacturers may freely pick monophage components from those listed in the dossier. So staying again in this example for the flexible product, this product would always contain 3 phages, but they can be picked freely from this list of 8 phages, and it's up to the manufacturer to do that. This freedom to customize the product is limited again to cases of development of resistance or changes in the epidemiology of bacterial pathogens in the field. So it has to be triggered by one of those two. But such customization actually would not require a variation application. So that could be done simply by the manufacturer. And in the gray box at the right, it's just stated that a practical consequence, for example, one of the practical consequences of this is that at any one time, different compositions then of this product could be actually used in different countries, for example, in the EU. Next slide, please. As regards development of manufacturing processes, and again, staying kind of talking about this flexible composition, the documentation required to support a flexible composition would obviously be minimized if manufacturing processes are robust towards this anticipated customization of products. So if a flexible composition is required, a good scientific understanding of the product and a good design of manufacturing processes would be expected. So this flexible composition option, it provides opportunities for manufacturers, but it probably also provides challenges. And some of those, again, are listed here in the gray box. So for example, the upstream parts of manufacturing processes may differ for the different phages which are listed in the dossier for flexible products. For example, bacterial host strains may need to be changed. So it may be a challenge to have processes which are actually sufficiently robust to maintain, for example, product purity for a flexible composition product. And also, for example, blending and formulation steps would need to be sufficiently robust to accommodate this flexible composition, not least, for example, as regards stability of the product and so forth. And again, this will need to be evaluated on a case-by-case basis, and if in doubt, there are scientific advice procedures available. Next slide, please. That was all about the quality requirements, actually. So now we are going to the part about post-marketing changes to products. So for phage products with a fixed composition, there may emerge, I guess one would expect over time, a need to update such products to address, for example, changes in epidemiology or resistance in the field. And even for flexible products, it may turn out that the flexible composition that was originally approved over time is eroded such that it's no longer sufficient to address, for example, changes in epidemiology or resistance patterns in the field. So for both types of products, fixed composition as well as flexible composition, post-authorization updates may be needed. And this will then require variation applications to the agency. Such changes in product might have a level of urgency, and so to provide maximal flexibility, we recommend in the guideline to the extent possible, formalize this with the agency in advance in the form of post-approval change management plans. Some details on this are provided in section 5, as mentioned, and also Annex 1 of the guideline. And so the purpose with such plans is to kind of try and talk through some of the critical issues involved in post-marketing changes beforehand. So when you come to actually making the change, people are on the same page and the process might be smoother. So examples of issues which might be addressed in such plans, for example, would be what would be the trigger for updating the product? So for example, if it's resistance, how will resistance actually be monitored in the field post-marketing? Which level of resistance is acceptable before a product update is triggered? When a product update is done, how will the new phage components be generated and so forth? These change management protocols need to be obviously realistic, so they need to be feasible and plausible. So they need to be based on scientific knowledge, understanding of manufacturing processes, understanding of product characteristics, the biology of the disease and so forth. And we obviously fully understand that for complex phage products, it may simply not be possible to answer questions such as those listed in the gray box. It may not be possible to make feasible and plausible change management protocols. But for some products, it may indeed be possible. And so where possible, these protocols might, again, increase regulatory flexibility. And so we recommend to consider this option. Next slide, please. And I think this would be the last slide on post-marketing changes. This is then just an example, I think, from Annex 1 in the guideline, where we kind of try to illustrate the different levels of complexity of updates and the different levels of quality, for example, but also efficacy and safety documentation that would be needed. We have here a fictitious example of a post-approval product update. So the approved product contains 3 phages. It's a fixed composition product. It contains phages A, B, and C. And now bacterial resistance has developed against phage C. So the manufacturer has trained phage C in vitro, has selected phage C in vitro to overcome the bacterial resistance, and now proposes to change the product. So taking phage C out and replacing phage C with the trained version of phage C, which is phage C+. And so the level of quality documentation obviously will depend on the overall complexity. So the simplest form of product update is shown in the green boxes. So for example, a simple form would be if the phage C and C+ are comparable, basically, obviously phage C+ has overcome the resistance, but if they're otherwise comparable, if this exchange of phage C by C+ is the only change in the product, if there are no changes to the manufacturing process, if there are no changes to the process control technologies, assays which are used to do control tests, if there are no changes to product specifications, then the quality data, obviously, to state the obvious, will be minimal. And then a more challenging, complex product update is shown in the yellow boxes. So for example, if this trained version exhibits significant phenotypic differences or biochemical differences from C, if the manufacturing processes need to be updated, changed significantly to accommodate this new trained phage C+, if there's, for example, a worsening in the impurity profile, such that, for example, even changes in product specifications have to be done, then you're almost talking about a new product here, and obviously the level of quality documentation would be quite high. So that is the kind of the general example. This is, I think, as far as we can go here until we actually get some experience with this type of products, with this type of updates. And the next slide, please. And this is the final slide, just to summarize. So as Susanna started by telling us, the general default requirements for phage products would be basically the requirements for veterinary biologicals other than immunologicals, but a flexible regulatory approach is expected for novel therapies. So the quality documentation, as I've tried to illustrate, can be adapted based on quality risk management principles. And as mentioned, we've tried to coordinate this with the pharmacopoeia, but texts do differ. So you need to consider both texts. And indeed, you need, when you read our phage guideline, to consider also other guidelines. And we've provided, obviously, a reference list in the phage guideline for other guidelines, which are also relevant to consider when developing your products. A flexible composition option is available for phage medicines in the guideline. The advantages is this rapid customization of products without any requirement for variation application. But the challenges are obviously that this requires a scientific understanding of products which can be probably biologically quite complex and very robust manufacturing processes. And then finally, if there's a need foreseen for post-approval changes, then if possible at all, try to formalize this beforehand with the EMA. And then to finish with the recurring mantra that this is a nascent field, so issues will have to be evaluated on a case-by-case basis, and do consider scientific advices during the development. That's all. Thanks.
Thank you very much, Martin. So I would like to open the floor for any questions or remarks that there are at this point on the quality part of the guidance. There is a question here in the room, so please go ahead, sir.
Does it work? Yeah. Yeah, the questions that we had, like we are Medea Biopharma, my name is Rüdiger Trojak. Question that we have is when you do the efficacy studies and you want to make a dossier with a large number of phages just to make sure that you have a good efficacy, it becomes a cost issue at some point to do the GMP production. And how to limit these costs, and how many phages did you have in mind for such a dossier? Like what was your idea when you wrote this guideline? What should be pre-manufactured? Is there any chance to list phages in the dossier which do not have to be pre-manufactured and integrated in the master bank to save the upfront costs? And how do I select my phages for the efficacy studies? Can I change the composition of the cocktail I'm testing throughout the study, or from one part to the from a study to another one?
So about the selection of phages for efficacy studies and composition of cocktails for efficacy studies, I think we'll come back to that in later part of the meeting during the presentation on efficacy and also safety. As regards the requirements for GMP manufacture, my guess would be that when you submit a dossier for a flexible composition product, for example, then I wouldn't think that you need to actually have a large GMP manufactured stocks in place of all the components of all the phages listed, but you would need to have characterized and validated master seed banks for all phages, and you would need to have shown that your manufacturing process is sufficiently robust to actually accommodate all those phages, so that you end up with a flexible product which stays within the same specifications, stays within the same, for example, stability, and so forth. Does that answer a bit of the question, or?
Yeah, can this be done outside of the GMP regulated environment, or does it have to happen under GMP conditions, this to prove the identity and stability?
There's a general requirement for GMP manufacture as such in the new regulation, but also it's considered, I think generally also on the human side, that for innovative products, this has obviously to be considered based on the product type. I think it's difficult to go into much more detail here, but what I can say is that I believe you may know this also, that the GMP regulation for veterinary biologics are in the process of being updated. And I believe that they will have a process similar to ours, where they'll draft a guideline quite soon and have it out for public consultation. And so it may be the best forum for you to try and get input in that forum initially, would be my suggestion, if that makes sense. And I think that the available information is available on websites. We can probably find that for you.
And if I may add to that, the GMP inspectors party have been made aware of this specific issue with the bacteriophages. So yes, we hope they will take it into account.
Yes, there are some questions in the chat as well. So I think, yeah, the first one was from Bob Reuter. I can either read it out, or he can also ask directly. So but yeah, I can. So the question was, would genetically modified medicinal products intended for veterinary use be obliged to comply with the requirements for contained use referred to in Directive 2009/41 ECEN, or deliberate release referred to Directive 2001/18 ECEN?
I think I'll say that we probably will come back to that in the part of the meeting on safety, and we can perhaps take that question there if that's okay.
Yes, I'm sure that our colleagues of safety will give us more light, but I will say that if the product is engineered and genetically modified, I will say that, yeah, that directive apply and that procedure should be followed. Yeah, I will say yes, but yeah, I'm sure in the safety part, we will have more information about that. Thank you.
Okay, so there are more questions in the chat. The next one is, sorry for the wrong pronunciation, Balash Kish. Is there a guide that deals with endolysins?
So that, I guess, would fall under, veterinary biologics, basically. And also, it would be relevant to consult basically ICH guidelines for biologics for inspiration. But they don't fall under our guideline as stated.
Okay, thank you. The next question is from Stefan Vermeulen. Regarding range, degradation of phages is a known issue. Will this be a no-go if, for example, phages go from 10 EE10 to 10 EE8? Or if you document that it might be acceptable?
So for all products, you have to state a shelf life, basically. So it would be up to manufacturers to state a shelf life where the product remains, where the safety and efficacy of the product is kind of maintained through that shelf life. I think is the best answer we can give here. So all products obviously decline in potency over time. And it's up to manufacturers to document that this during the approved shelf life does not impact the safety and efficacy, if that makes sense.
Okay, thank you very much. The next question is from Barbara Brenner. Is there an option foreseen for emergency treatments like acute infections, which will need treatment on the spot?
I think here, I think I'll refer to you, if I may, Jean-Paul. Is that okay? Because emergency treatments would be difficult with a centralized product, you could say. So if you have an emergency where a centrally approved phage product doesn't fit, then you wouldn't have time even for a flexible composition, probably. Well, you may have time for a flexible composition to update the product. So actually, for a flexible composition product, if you have an emergency and some of the phages which are registered in the flexible composition work in that emergency, then the manufacturer can basically update the product and treat the patient on an acute basis with that product if they have luck. And if they don't have luck, meaning that if this emergency is not covered by the flexible composition, then they would need to use magistral phage preparations. And perhaps, Jean-Paul, you would like to say a word on that, or?
Yeah, I think I have more or less the same answer. I'm looking forward to the presentation of EDQM, because I think that, I hope, will give a solution for the more flexible approaches.
if I may, for veterinary product and for extemporaneous use and magistral product, the rule is the cascade, which has been updated with its new regulation and which is more difficult to apply. And so we need to have interpretation how to apply cascade for phages. Thank you.
Thank you very much.
Yes, please go ahead.
Can you hear me? Okay. Good morning, and thanks for the opportunity. In the document on page number 9, if you see line 277, there is a group of coding sequences that are not allowed or are not expected, but in some cases it's impossible to find a host without those genes. You allow, for example, an exemption in the case of prophages, but in antibiotic resistant genes is common to have those genes in the host for some very specific clinical or for specific diseases. When you have just one etiology agent, you cannot find a host. So do we have the opportunity to use PCR, for example, to detect or to provide evidence of the absence of these genes in the product as an alternative?
As an alternative to what?
To the absence of the genes in the host bacteria. I mean, can we manufacture in a bacteria with these specific genes if we can guarantee that the genes are not present in the final product, for example?
Yes, I would think that might be acceptable indeed, and for bacterial hosts, for characterization of bacterial hosts, certainly PCR assays would be one of the type of assays that could be used, and also for control of products, obviously, and this also is one of the aspects that would be specified in the general chapter of the European Pharmacopoeia. So you would have both our guideline and the compendial text to look at in this situation.
And do you expect to have the phages in a deposit, for example, the ATCC or something like that?
No, that wouldn't be a requirement. So these obviously could be, you could say, a company property, and there's no requirement as such to share these commercial products.
Okay, thanks.
There is another question in the room. Yes, please go ahead, sir.
I actually had the same question with the previous one. In the drafts, it stated that the host on which the phages are propagated should not carry these unwanted genes like antibiotic resistance, but also virulent genes like toxins. How is this possible? We are talking about phages for pathogens. All pathogens have toxins. So what's the point? Have we understood something differently?
No, I think not. So I think the general requirement is that the materials used for manufacture should be as clean as you will as possible, but I think it's hopefully also clear in the text that this is evaluated on a case-by-case basis. So if, for example, you have a host bacterium which actually does carry, let's say, a toxin gene, but where you can justify that this does not give any kind of risk for the final product, then my guess would be that if that is justified, if appropriate process controls are in place, then that might be acceptable. Jean-Paul, you look like you want to add.
Yeah, I think I agree that this needs to be looked at a case-by-case basis, because virtually everything that a pathogenic bacterium produces can be seen as a toxin, but there are differences. You have Shiga toxin, cholera toxin, whatever. And I think also like the combination of these genes with prophages, for instance, I think therefore, I think it should really be looked at a case-by-case basis.
And again, to add here, you will have, and I think this is an advantage, you will have this advantage here to have both our text to look at, our guideline, and also the pharmacopoeia text. And so based on those two, and obviously based on the normal interactions that you would have, the normal regulatory interactions during product development and submission of dossiers, these would be issues that would be discussed and I think in most cases reasonably addressed, would be my expectation.
Thank you for that. I think there are further questions in the chat.
Yes, and there was also just one hand raised, but I don't see it anymore. It was Miguel Campanero. Do you still have a question? Okay, that doesn't seem to be the case. Then we have another question, again from Stefan Vermeulen. What is comparable? Is there a certain cutoff of, for example, percentage of mutations? And then again, directly afterwards, he asked, what is comparable? See example slide. Is there a certain cutoff? Okay. It was just a repetition of the question. For example, percentage of mutations.
So this comparability question goes to the issue of post-marketing product updates. And we provide in Chapter 5 and Annex 1 more text on that. It may be difficult to go into detail here, but basically comparability, the term comparability that we use in our guideline reflects an ICH guideline, which is also referenced in the phage guideline. And no, there are no fixed cutoff levels for comparability. But nevertheless, this concept of comparability, we think can reasonably be used, is actually being used in connection with, for example, product updates or biosimilar products. So again, I think it may be difficult to go into much more detail here, apart from saying that please look at Part 5 and Annex 1 in the guideline. And if you feel that there's something there which doesn't make sense or you have additional questions, then please send us our comments or suggestions, as Jacqueline and Susanna mentioned, here after the meeting, would be my suggestion. Thanks.
Thank you. There are two more questions in the chat. Again, from Barbara Brenner. Does the guideline apply to phages manufactured in clinics for individual use on the wards?
No, the guideline, as Susanna stated, applies to centrally authorized products. So for medicinal products, this guideline would not apply.
If I may, yeah, this guideline applies to industrially manufactured bacteriophages. So when bacteriophages are manufactured like a magistral formula or only for a one patient use, then this guideline doesn't apply. only for industrially when you expect to produce several batches, and well, more like a medicinal product, veterinary medicinal product that we find in the pharmacy, but not for individual use, and that's prepared only for one patient. Thank you.
Thank you. And the last question in the chat so far is from M. Stanchik. Does EMA provide more clarification on the methodology we can employ to determine the host range of bacteriophages?
It's not I would say the agency's task to do this. This is up to manufacturers to do. But I think we are in the fortunate situation that there's a wealth of scientific literature dealing with this, and we actually provide a lot of citations in the guidelines. So this is something that manufacturers need to do, as for any product, during product development, to perform the appropriate assays on the appropriate pathogens, taking into consideration the type of product, the product profile that they have in mind would be my answer to that. Thanks.
Okay, thank you. We have reached the end of the questions in the chat. If there are any further questions at this point, please raise your hand. If not, then we have made it to the coffee break exactly on time. Well done, everybody. Thank you all for the discussion so far. And we will see you back at 11 o'clock for the next presentation, which is on the safety part of the guideline. Thank you. Welcome back, everyone. I hope everyone is back, at least here in the room. We are again complete. And the next presentation on the safety aspects of phage therapies will be by Sandra Maria Wienhold. She works as a scientific assessor at the BVL in Berlin. And she's also a member of the operational expert group on bacteriophages, and she was responsible for the safety part of that. So please go ahead, Sandra.
Thank you for the nice introduction. So I'm speaking here on behalf of many experts who provided all their knowledge into the guideline. Especially, I would like to thank my colleagues from the BVL and also from the UBA, the German Central Environmental Authority. Next slide, please. So as said, I will focus on the safety part, and also here we have the safety documentation and some general considerations. And yeah, the documentation shall be presented in accordance with the Annex 2. As we already said, there is some guidance also for phage products, and the CVMP and IICH guidelines are also applicable. The requirements of the annexes and also of the guidelines may be adapted if it's scientifically justified and based always on the required specific product properties. All the risks, so here especially all the safety risks, should be proactively identified by the applicant, and therefore a risk profiling methodology should be applied, and also the quality risk assessment stated in the quality part should be taken into account. If safety risks cannot be excluded, control and mitigation measures can be applied to reduce the risk to an acceptable level. In the safety part, and especially for veterinary medicinal products, we have to take care of the consumer safety, which means that food-producing animals are of special concern. For these kind of animals, withdrawal periods have to be established, even if the withdrawal period is 0. And this is specific for a formulation and also for a dosage regime. And to set up this withdrawal period, maximum residual limits have to be defined, and these shall be considered in accordance with the relevant regulation in advance. It's important that here the active substances and also the excipients are considered, and also, of course, the target species, because it's a difference if you produce a product for cattle, for instance, or for honeybees. And of course, also the relevant tissues and products of the animals should be considered, which is milk, for instance, or also honey. The agency should be consulted for the need of an MRL evaluation in advance. Next slide, please. We had a lot of discussion about this topic in the group, and finally, we defined or set up these 2 positive outcomes for the MRL assessment. So first of all, the phages or the active substance of a specific product could be included in the list of chemical unlike biological substances, which are considered not requiring an MRL evaluation, following the Annex 1 of this specific regulation mentioned here. Or, on the other hand, it could be possible that the active substances are included in Table 1 of the Annex to the Commission Regulation 37/2010, which means that an MRL need to be established and is set. The excipients, as always, can be included in the regulation I mentioned before, so an MRL should be established, or they could also be classified as not falling within the scope of the regulation 470/2009, which means that also here no MRL is needed. Next slide, please. So coming to the safety test, the safety for the veterinary medicinal product means user safety, environmental risk assessment, and also target animal safety. And here also the Annex 2 is applicable. The target animal safety part is also, it's only mentioned partially here in the guideline in the part 3, because we decided to put all the details for the target animal safety studies in the efficacy chapter, so this will be discussed later on. All the studies, including toxicology and also special studies, could be carried out with a representative monophage or multiphage preparation. We already heard this in the quality part, and in regard to the safety tests, we defined that these representative preparations should represent a worst case scenario. So to say, they should contain, for instance, the maximum amount of protein, or the maximum amount of phages and the maximum possible titer, so that we can see all effects with this representative preparation, and do not have to see it in the end with all different preparations with less titer or less protein. Next slide, please. For the safety test, the extrapolation is possible between comparable strains of bacteriophages. We also already discussed something about comparability, so this is also a case-by-case decision then here. It is possible to extrapolate between target animal species and different route of administrations. This extrapolation should be based on representative and validated in vitro or in vivo parameters, or in well-justified cases could also be done on literature. Next slide, please. For the precise identification of the product, here especially for the bacteriophages, the requirements of the Annex 2 might not be adequate or are not adequate here. So this is here also a part linked to, especially to the quality part. The identification of the active substances should be tailored for viruses and based on the requirements set in the quality part. And here in this part 3A1, the formulation of the product should be stated and should be, of course, in line with the point 2A1 of the quality part. Next slide, please. For the pharmacological data, these data are, as always in a veterinary medicine application, necessary to provide data of the fate of the phage, and to characterize the mechanism of action and pharmacodynamics relevant here for the safety evaluation. In terms of 3R principles, we state that the absence of studies in laboratory animals could here be justified by referencing existing data or data from the target animal studies. Next slide, please. So we know that phages only replicate when the host is available. Therefore, conventional absorption, distribution, metabolism, and excretion studies may not be appropriate, and in most cases, they are not possible. However, this data is necessary so that safety could be assessed, and but all the data could be drawn from studies in the target animal species. So we do not need special laboratory studies here and special studies out of the target animals. But these studies have to be adequately designed and to address the evaluation of the safety. Information which is needed is the absorption from the site of administration, dissemination of the phages to other anatomical location, and also expected degradation pathways. To conclude on the fate of the phages inside the animal, we set two prerequisites. So first of all, the phage should be studied in target animals without the active bacterial infection, so that the absorption could be studied, and then of course the distribution and also excretion degradation should be studied when the product will be used to treat animals in the presence of the host bacteria. Here, the data should be derived from appropriate sources, which could be dedicated PK/PD studies, in vitro models, pilot efficacy studies, or a combination of all of these. We set here an EG, so we are pretty open for any kind of studies which could address the safety here. And as we already heard this several times during the meeting, it's not possible to give exact guidance here, so it's a case-by-case decision because the products are novel and also very complex. Next slide. We also know that bacteriophages do not propagate in eukaryotic cells, so a direct effect on the animal or also on the human is pretty unlikely, so there is no mechanism-based concern for toxicity in the target animals or in the humans. However, we all know that there is the potential presence of contaminations which are endotoxins or exotoxins, and this is considered a safety concern also in human medicine, of course. And the control of these aspects is very essential during the manufacturing process. So also here, quality should be assured and can be sent in also in the safety part. In agreement, again, with 3R principles, we decided to that single dose toxicity and also repeated dose toxicity, and as well as the effects on reproduction and developmental toxicity can be addressed by the controlled manufacturing process. So no special studies are needed here. The target animal safety studies can be used to address this issue, and also literature data according to current state of the art. So you may not send in the papers from the 1920s, but new literature. If a specific safety concern is identified, supplementary studies might be submitted here. As I already said, there's also no direct interaction with DNA or chromosomes expected, and also no influence on eukaryotic cell proliferation or immunosuppressive effects. So we decided that the range and type of genotoxicity studies which are routinely used and needed for pharmaceuticals are here not applicable, and that the standard battery of genotoxicity tests can be omitted for these kind of products. And also carcinogenicity studies most likely are or can be omitted as well, but it should be addressed in the dossier, of course, just with a few sentences. Coming to the special studies, we have to take into account immunogenicity and immunotoxicity of these potential products. And also here, we do not ask for special studies. The data could be drawn from the target animal studies again, combined with the proposed posology of the product, and also on existing knowledge on immunogenicity and immunotoxicity of the phages which are used in this special product then. But for veterinary medicinal products for which skin or eye exposure may occur, we have to state that the general requirements in Annex 2 have to apply. Another part of the safety requirements is the development of resistances and the related risk to humans. As I already stated, a direct risk from bacteriophages to humans is very unlikely because they are all around us in our gut and our skin. So here specific studies are most likely to be omitted. But a big problem, and I think we are all aware of this, is the resistance development. Over time, bacteria will develop resistance against bacteriophages, and the applicant should reflect upon this risk, or not on the risk of developing resistance, but on the release of these resistant strains into the environment and the related risk to humans associated with the use of the product then. For the user safety, the user is everyone which handles the product before it's applied on the animal or during the application. There is no specific guidance available for biological products other than immunologicals. But specific warnings have to be included in the product literature, and to derive these appropriate warnings, general principles on user safety as lined out in the mentioned EMA guideline should apply for phage products as well, and should be taken into account when an application is sent in. The information obtained from the assessment and the hazard identification will be considered for the risk characterization, but in cases where no information on dose relationship is available, a qualitative risk assessment might be also sufficient. So now we're coming to another part which we discussed a lot in the group, which is the environmental risk assessment. I think this is something which is not so far as I know, addressed in human medicine, but for the veterinary medicinal products, it's very important because, of course, these products will be used on the food-producing animals, and these bacteriophages will enter the environment by direct excretion or also by application of manure to the agricultural land. There's only limited research on phages which are, so to say, released by humans and not native to their receiving environments. So there are a lot of uncertainties about the fate and effects of such bacteriophages in the environment. There's few experimental studies which indicate that changes in the microbial community composition occur if phages are released and that the natural ecosystem is altered in some way. Therefore, the applicants should reflect upon the environmental impact to soil bacteria and soil function associated with the use of the product. And these tests should be in accordance with the OECD guideline. And for instance, OECD guideline 216 could be taken into account. Here we have the part for the genetically modified bacteriophages. We included them here, so we say they are genetically modified organisms and therefore should be handled according to the chapter of the annex and also relevant guidelines. Next slide, please. So now in the last slides, I will also follow Martin and talk about the post-marketing authorization changes. I also want to give some general remarks. The Annex 2 already recognizes that phage products will need to be updated on a regular basis, which is pretty new and gives a lot of flexibility, but it's also difficult to give a guidance here. As Martin already said, the authorization of these updated phage products can be streamlined based on studies with the parental product combined with other relevant data. And this data could be derived during the lifespan of the authorized product. So for instance, pharmacovigilance data could be used, or the improved understanding of the manufacturing process, and of course also every knowledge which was gained during the lifespan of this product. But of course, the scientific standards for all of these data is the same. So it should be demonstrated that the proposed update will have a positive effect. So for instance, restored efficacy without negatively impacting the quality, safety, and efficacy of the product. As I already said, it's not possible to specify exact data requirements here. As already Martin said, it's recommended to consult the agency on a case-by-case basis. But also here we have this comparability, and we state if the parental product and the new product are totally comparable, then there's no need for additional studies on quality, safety, and efficacy. If there is a relevant level of difference between the two products, additional studies might be required. But for sure, if the comparability is significantly different or it cannot be concluded at all, how the products or how comparable the products are, then in vitro studies and also in vivo studies will usually be required. Next slide. So talking about the updates, Martin already explained the nature of these updates. We have the simple updates, which means that there's addition of a monophage, which is very comparable to a component which is authorized with a marketing authorization application. And here, safety studies are not expected and not required. These post-authorization changes are approved then on quality data alone. In the same line, user and environmental risk assessment is not expected to be required. And coming back to the MRL status, the EMA needs to be consulted here. And next slide. For complex updates, it's, as we already said, safety studies might not be required if the impurity profile of this product is not substantially worsened. Also here, it's a case-by-case decision. In high level of complexity or lack of alternative evidence, safety data may be required, and a new user risk assessment and environmental risk assessment might be needed. And again, of course, it is advised to consult the agency for the MRL status. So I think this was the last slide. And I want to thank you for your attention, and I'm open for discussion.
Thank you very much, Sandra. I'm looking to my left to see to you, to see whether there are any questions already in the chat. Oh, I'm sorry. I see a hand to my right. Please go ahead, Dr. Boersma.
Thank you very much. Thank you very much for this very insightful presentation. I have a question on the MRL, whether that's needed or not in combination with the withholds. And it said that most probably the phage products should have a withhold, even if that could be 0 days. But how would you establish a withhold if there's no MRL? And how, yeah, that's my question. And in which cases you think an MRL would be needed?
Yeah, that's a very, it's a great question, a very tough one. So we had, as I said, a lot of discussion, and this is the current state. I don't know if Susanna could add more, but I think this is what we, in the regulatory framework, had to write there and should state there, and we will see how it went when we have the first product, maybe.
Yeah, but for the commercial potential of a product, it's very important to know in advance if there is a withhold required. So how would we need to assess that?
Yeah, what we state in our guideline is we advise the applicants to consult the EMA, because there are guidelines applicable to maximum residual limits for biologicals that when drafting this guideline were ongoing. So we couldn't go further. That's why in the guideline we state that at the moment you plan to register your product and before, of course, submitting the application, then to consult the EMA. Because we for this matter we need to align with another regulation, and sometimes the timelines are not the same. So what we can advise at this moment is that you need to consult the EMA on this matter. Thank you.
Thank you very much.
So there are again some questions in the chat, but I think they are still leftovers from before the coffee break. But since there are no other questions, I think we might go forward with them.
Well, yeah, there was the one question on the GMO point, I think. So I guess that it falls under 2001-18, but maybe there is a comment from the panel on that.
Yeah, I also checked my notes, and it's always said that the Directive 2001/18 on deliberate release into the environment of genetically modified organisms should apply here for the phage products.
Okay, so we have a question from Hans Kleppen. He's referring to line 281 to 282. The detection limit of PCR-based techniques as a threshold for amount of excised prophages has no biological relevance for the quality, safety, and efficacy of the phage product. If such a technical threshold is set, genetic modification of production horse to inactivate prophage induction must be considered. It would be better if the guidelines stated that the maximum amount of excised prophages needs to be justified on a case-to-case basis.
I think I can take that. I think I can only say thank you for the excellent observation and feedback. And if I may, please do supply this comment, if you can at all, in writing to us. And then obviously we are still in the consultation phase of the guideline, and I'm sure that many oversights can be found. So please do supply that comment to us so we can take it into consideration during the drafting of the final text. And again, thank you for the observation.
Yes, thank you very much. That's indeed a very good point. So everyone, please, also from the EMA side, again, provide your comments in written as well. That would be really helpful. Thank you very much. And there's another comment directly to the guideline from Barbara Brenner. As of line 264 of the draft, absence of lysogenic activity. Impossible to prove. Proof of lytic activity is sufficient to exclude lysogenic activity. Isn't genome sequencing sufficient to exclude the existence of lysogenic activity?
Sorry, I think again it becomes very complex to answer. I'm not actually sure that I'm even qualified to give a full answer to that question, but I think I would refer again to what's stated in the guideline and what's also stated in the pharmacopoeia text about characterization of phages, and then end up with the general observation that through the guideline text and also as regards this characterization of phages with regards to lysogeny, I think we do state that it's a case-by-case evaluation. And so it's obviously up to manufacturers during the normal regulatory interactions to provide their case, their justification for the product. And then, at least my experience is that even for complex products, a way can be found, if that addresses the question. You look like you want to add something, Jean-Paul. Please go ahead.
Yeah, I would just like to add that I don't think that when you see lytic activity, this means that you do not have a temperate phage. And I think that when you look at the genome, you can screen for known determinants of lysogeny. I think that's possible. Of course, only what is known, but that's already a good starting point, I think. And indeed, it's a case by case. It's the same thing for the prophage. It depends also on which prophage you have. So it's very complex and case by case, I think.
But specifically to this issue about genetic characterization, so there again in Annex 1 of the guideline, some details are given for that. So that hopefully is also of help. And again, if you feel that we've omitted something as regard to genetic characterization, then please do submit comments to us, and this would be very gratefully received, because this is a complex area. We can use all the help we can get, and we'll take that into consideration during drafting of the final text. Thanks.
Thank you. There are still several questions in the chat. Perhaps I will continue now with the last question because this is safety related. So MRL, it's from Avner Finger, I hope I pronounced it correctly. Using natural phages in product will trigger requirement for MRL. This requirement might reduce dramatically the commercial interest in the technology.
Yeah, as we said, we have to, for food producing animals, MRL needs to be taken into account, and we have to set this, or have to establish this section here. We will see, as Susanna already said, how it's going with the other guidelines and see what about what's happened with the biologicals and the phages here.
And also, if I may add something here, and please just correct me if I'm wrong, but it seems to me that this issue about MRL and residue limits is something that at least can be addressed at a very early stage of development. So the information that you need to evaluate with the agency whether MRLs are needed, you hardly need to have done any experiments at all. You just need to have like a general product profile. And then based on that, you can get an answer would an MRL be needed or not. So it's not like you need to invest millions and millions before you get an answer to this. You can get this upfront, and then you know where you stand, would be my observation at least. Thanks.
Dr. Scafley, please go ahead.
Yeah, thank you, Jacqueline. Maybe just to further clarify the need for an MRL and the need for a withdrawal period. Each product needs to contain a withdrawal period on the SPC. It can be 0 days, it can be several days. That is depending on the safety of the product. The need for an MRL is it's not an obligation because the definition of a withdrawal period is that it is the period between the last administration of the product and the time that the residues are safe for the consumer. It's not directly related to the MRL in the legislation. So if you can, for your product, justify that the residues will be safe, and that would be enough to obtain a zero withdrawal period. But for the need of an MRL, it is advised in this particular guideline to consult the EMA, because if an active substance does not need an MRL, this needs to be listed before the application. So there needs to be a list of substances that don't need an MRL. Otherwise, you need to have an MRL. So those are the two options, and for that you are advised to consult the agency before the application. So there's certainly not the intention that all the bacteriophages need to undergo an MRL assessment. That's certainly not the intention of the guideline. The only thing that it's not, this guideline does not provide the final answer. And that is only because there was still another guideline in development relating to the MRL, the need for MRL for biological substances. And it was not possible to provide further guidance in this particular guideline because the other one was not finished yet. So, but just to make sure that there is no strict requirement to undergo an MRL assessment, and that you are advised to consult the agency before you develop your product further. Thank you.
Thank you very much for the clarification. Perhaps I can directly come to the question that just popped up. So why not to include natural bacteriophages as a group in out-of-scope list for MRL?
Well, if I may, I will try to answer this question. We discussed previously on this, and we didn't find the pathway to do that So as Dr. Sefferli said, there is a guideline on development now applicable to MRL in biologicals. And we are, I think I can say we are working on it, but by now we can only say that the EMA should be consulted. We try to explore that possibility, but at the time that we were developing the guideline, it was not possible. Thank you.
Thank you very much. There's okay. There's another question. Is there a recommendation for a number of patients to be treated in order to confirm the safety profile?
No, we do not set a recommendation here. Could be 2 patients or animals or 10 or 20. So we will see what we will receive as it's depending on the target animal safety studies. There are recommendations there, of course, but no special recommendations for the safety here.
Okay, thank you. There's another question regarding MRL. Will phage be considered as protein plus DNA only, or will MRL assessment also have to take into account different properties of phage with different genetic content, natural variants and or genetic modification?
Well, we don't have an answer to that, because the establishment of an MRL and the evaluation of the needs of an MRL is out of the scope of this guideline and also of the Regulation 6/2019, because it's under another regulation. So we don't know what will be the considerations on that. And I want to add also that we are really new on this kind of products, at least in the regulatory point of view, and we are trying to establish a starting point. And for MRL, it's the same. So I think we need to work more or less together and try to adjust the regulation to the need of the developers, and also the developers to collaborate with us. So for MRL, it's a totally new field, and it's the same for the other part or requirements of the regulation. So for MRL, as we, I think, said before, please consult the EMA, and of course we will need a case by case analysis, and because of the characteristics of these products that are entirely new and we don't have experience, this is the starting point that we establish. Of course, the guideline will be reviewed, I think, in the future, if needed, when we have more experience. But by now, we try here at least to start and to prompt the development of this product. Thank you. Sorry, I think Dr. Pirney want to say something.
I would just like to add, like, a personal scientific advice is that I would also be in favor of a case-by-case approach, because I think, for instance, if your residue is like active phages that, for instance, would target E. coli, that could have also an impact on consumers. So, but it's really case-by-case, I think.
Yes, please go ahead.
So I have a question. How do the inspectors will assess the residues in meat products or in animal products of phages? And how can we say that the phages that will be detected are the phages that we have administered and not naturally occurring phages infecting the same host. So how are we going to differentiate at the level of inspection?
I can at least start by saying something and to start the discussion. So I think the issue, I guess, starts during the development of the product, where you need to figure out whether a withhold time and MRL setting is relevant for your phage. And there, obviously, this assay question is easy because there it's done in controlled animal studies under controlled conditions. But I think your point is very well taken. If you have a phage product which is 100% identical to a naturally occurring phage, then I guess the only one of the ways that could be used is, for example, if the natural phage doesn't occur in the same anatomical location as your phage product, that could be used. So even though your assay can't discriminate, then perhaps if it's a different anatomical location based on administration, that could be used during inspection. Also, even if assays are not discriminatory, then levels of phages could be used, meaning that you would have like a knowledge about the normal background level of that particular phage. And then, let's say in an abattoir, if you find levels which are dramatically higher, that could be ascribed, for example, to use of the product. But this certainly, assay-wise, might be a challenge during inspection, of course. Thanks.
Yes, there's another question here in the room. Please go ahead, sir.
I have a comment and a question about the definition of concentration of phages, because it will impact safety and MRLs and stuff. Let's start with the comment. And in line 302, on page 10, there is potency, and you declare that potency is expected to be technical possible to be determined for each component in a multi-phage formulation, but that is not real in some cases. For example, if you have preservatives, or if you have 2 or more phages produced in the same host, so in some cases you won't be able to determine the potency. So can we use different methodologies for concentration? And the second one is, what happens if the potency changes dramatically when you include a new phage, because the relationship between enumeration and potency changes a lot between phages? Thank you.
So the question that you asked relates to characterization of the products, and this general requirement that for products containing more than one phage, we would like to see potency estimates. That means that the biological activity of each phage component, we think is important, because I think it's expected that, as you said, the stability, for example, of products such as this, phages can influence each other, that can aggregate. So it's difficult in multiphage products, if possible at all, to have potency assays for each phage components. We do, however, also recognize in the guideline that it may not always be possible For example, if some of the phage components use exactly the same bacterial host, and it is also stated here in the table that you referred to, that in such cases alternatives might be considered. Again, it would be up to manufacturers to justify this, but I think this requirement for potency assays for individual components is quite important. And there really would need to be very strong justification for not doing this, and also an understanding what impact this has on, for example, product stability. And I think you will find the same philosophy in the pharmacopoeia general chapter also. So it is an important requirement, but obviously one that, if justified, might be tackled somehow differently, if that answers your question. Thanks.
Yes, it answers. Thanks.
So there are still 3 questions left. One is more general and 2 are, I think, more related to quality and not safety, but we still have some time, so we can go forward. Okay, so the general question is, how can the European vets contribute to the development of those products? Has any plan been considered to involve more doctors in the EU?
If I can start the discussion, I would say that what I would like to see is from practitioners ideas for applications, like actual medical problems that they feel could be solved by phage products, also by simple products where we could get this experience that Susanna talked about, with simple products that may be easy to approve and actually solve a real medical problem in the field in large-scale populations would be my input. Thanks.
Yes, and to add to that, obviously the Federation for Veterinarians in Europe is always included, I think, as a stakeholder in all our communication. So they are welcome to comment or provide suggestions in any way. Thank you.
Okay, so there's a hand raised. I see Hans Kleppen raised his hand. So please go ahead.
Oh, with sound. So I have 2 comments. One is to the question earlier on potency. I think potency is the most important quality attribute of a phage product, and it has to be documented. If it is impossible to document it in a mixed product, I think maybe a solution where you produce the phages individually, you store them individually, and then you mix them just before administration, or some other way of differentiating them during quality methods. But I think as was stated also, that it should be really good reasons for lowering requirements on potency. So that was one thing, and the other was a comment to Pantelis's comment. Both Pantelis and myself work with phage in aquaculture, where a typical effective dose of phage in water constitutes about 10% of the naturally occurring phage, and it is going to be impossible to differentiate between the added phage and the naturally occurring phage with respect to MRL assessment. And I think EMA should take into, maybe as part of this guideline or the ongoing work on the new MRL guideline for biologicals, take into account or some sort of yeah, some advice on how MRL or the lack of a need for an MRL could be assessed in an effective and easy way.
Okay. So personally, I think that the actual potency of the product is very difficult. It's a difficult issue, and it's really like based also dependent on the bacterium that you target. You can determine an activity in your lab, but the real activity must be seen on your animals, and there I think the animal study is important. And then for the residue, I think, for instance, when you target fish pathogens, I don't think there's a real problem for humans there. But if you, as I said, if you would target like certain E. coli strains, there might be an issue because also these E. coli is also present in the human microbiome. So that's why I think a case-by-case approach is really the best way to go.
I think Dr. Schäffli would like to react.
Yeah, maybe just to say that there's a lot of interest in the MRL area. I hear that, and I can understand it. But the reason why it's not specified really in the guideline is because there's another guideline in development, and it's the way it's stated in this guideline is not because we have particular safety concerns with bacteriophages in general. It's just more a regulatory issue on how to address it in a way that that is in compliance with the legislation. So to make it, to solve that in a regulatory way, compliant with the legislation, that's more the issue than for a particular safety concern for most of the bacteriophages anyway. So just to say that, so this guideline is not intended to address that particular topic because there's another guideline in development for that.
Okay, thank you very much. There are still 2 questions left. So one again from Barbara Brenner. I'd like to verify again on my previous question on magistral production. The EU GMP guideline, which is still in force, states that the principles of GMP are also relevant for manufacturing processes, such as that undertaken in hospitals, part 3 of the introduction. Will this be omitted then in the future?
It's not a question we can ask. So the question relates to whether GMP applies to magistral preparation of phages. It's simply not within the scope of the guideline we are discussing today. So at least from my point, I would need to refer you, for example, well, basically to, I don't know, look at the actual guidelines. Perhaps, I don't know, can regulatory colleagues give an indication about where one could consult for more knowledge here?
Thank you. And there's one last question from a participant who unfortunately had to leave the meeting, but still wanted to bring up this question here. For the discussion, I would like to share with you an important comment regarding lines 276 till 282 of the guideline in relation to paragraph 51544 of Annex 2 concerning the absence of genes coding for known virulence factors. The degree of mandatory use of production strains without such genes should be considered with great care. For example, in Pseudomonas aeruginosa, the endotoxin, or LPS, is a receptor for many phages, and it is therefore impossible to produce these phages on strains lacking LPS, which is also a major virulence factor. The same is true for certain membrane proteins that are antibiotic efflux pumps and phage receptors. Perhaps this type of exception should be explicitly stated in the document so as not to limit possibilities. Similarly, it is possible that a therapeutic phage could only be produced on the pathogenic strain of the patient. What is your position on this?
Thank you again for the comment, and I think we had a similar question before. So again, we certainly recognize, and this was also up in discussions during the drafting of the guideline, and I think it's also been up in discussions during the drafting of the pharmacopoeia text, that obviously it may not always be possible to produce phages, for example, on host strains that are free of, let's say, toxin genes or other virulence factor genes. And the text that we've adapted here, I think it's in line 279 of the guideline, is that if you can't have a completely free production strain, bacterial production strain, for example, as regards prophages or toxin genes or whatever, then it should simply be justified that whatever is in your bacterial strain, whether it's prophages or toxins or whatever, that this has no detrimental effects on the safety and efficacy of the product. And I think in most cases, this can be done relatively simply by appropriate process controls, where you simply show that in your final product, the level, let's say, of bacterial DNA or bacterial proteins or whatever is below the required threshold. So it's something which can be addressed, I think, in practice, and the guideline text, I think, to some extent, does that already. Perhaps we should stress that even more. But I think the principle that we follow in the guideline, that the default thing we ask for is that, for example, manufacturing strains are as clean as possible. I think that's the correct way to go. So to say, that use the cleanest, for example, production strains that you can, and then if they're not 100% clean, which they probably often will not be, then address it by common sense approaches, by appropriate process controls and setting of specifications for the product. Thanks.
Indeed, and when we talk about worst case scenarios for representative phage products, this is one of the things to consider. Yes, I see nodding. Did we have one further question? No, that's all. Okay, then if there are no further questions, again, we are perfectly on time for the break. So we will stop for a lunch break now. And I would like to see everybody back at 1:15, 13:15, back in the room. Thank you. So I hope you all had a pleasant lunch break. Apologies for being slightly late here. There was a huge queue for the coffee. But anyway, we will get on with it. The next presentation will be on the efficacy aspects, the efficacy part of the guideline. It will be presented by Belén Guitterier. She is a assessor also at the Spanish Agency of Medicines and a member of the operational expert group responsible for the efficacy part mainly. Belén will be presenting from Spain. So I hope the IT facilities will help us here. So I would say, Belén, go ahead.
Hello, can you hear me well?
We can hear you well.
Okay, thank you, Jacqueline, for the presentation. And so I am going to explain the different sections of the guideline that include efficacy requirements for these products. And I also want to thank all my colleagues there that contribute to the guideline and all the people in general that has contributed to the guideline. Next slide, please. So I will, in this presentation, I will address section 4.4, which include efficacy requirements to the parental products. Also section 4.5, 5, in which we have taken into account the possibility of a concomitant use of bacteriophages with conventional antibiotics, and Section 5 and Annex 3, which include efficacy requirements for post-authorization updates made to the phage products that Martin and Sandra had already mentioned, that it could be made to overcome bacterial resistance or to address changes in the epidemiology of bacterial pathogens in the field. First of all, regarding, next slide, please. So first of all, regarding the efficacy documentation, the same as for quality and for safety, general requirements for this marketing authorization laid down in the Annex 2 of the Regulation 6, 2019 for biological medicinal products, other than immunologicals. Also, the CBMP and the BITS guidelines concerning efficacy are also applicable. And they are acceptable, adaptations to all these guidelines and could be and to the general requirements could be accepted, but control and mitigation measures to ensure that the risk remains acceptable should also be proposed. So in general, a full efficacy package should be provided for the representative monophage or multiphage preparation. And then the extrapolation of this efficacy for alternative combination based on validated in vitro or in vivo data or scientific justification could be possible. So generally, efficacy and safety should be demonstrated by studies in the target animal species under laboratory conditions, that is the preclinical studies, and then supported by field conditions. Next slide, please. So the efficacy documentation will include preclinical studies containing pharmacology, the development of the possible development of phage resistance and related risk in animals, dose determination and dose confirmation studies, and tolerance in the target animal species. Then clinical trials in which we have special considerations for metaphylaxis and prophylaxis claims. And then again, we have taken into the consideration the possibility of the concomitant use with conventional antibiotics. Next slide, please. So normally in the preclinical studies, they are performing target animal species. However, the omission or replacement in non-target animal species or in vitro data may be possible if sufficiently scientifically justified. So the preclinical studies support the use under the recommended conditions, that is the route of administration, the dose interval, the resistance. And in general, target animals are used for the representative monophage or multiphage preparations. These representative preparations should be justified considering the indication. And then in vitro data for alternative combinations are possible with a scientific valid extrapolation. Next slide, please. So pharmacology covers the mode and mechanism of action, that is to demonstrate that they are lytic and they do not contain genetic determinants that confer lysogeny to the phage or virulence or antibiotic resistant to bacteria. This has already been discussed a little bit previously in the quality part, and some questions have been posted in the tables. So all this information is in the quality documentation. Then the range of hosts should support the claims. For this in vitro test could be used, and the activity against target pathogens and non-targeted bacteria should be addressed. And non-targeted bacteria, for example, in the case that these phages affect the commensal bacteria, for example. And the isolates will be clinically representative of the strains found in the field. So regarding the posology, it is suggested to demonstrate that the recommended dose and dosage and the administration route of the representative monophage or multiphage preparation results in a productive bacteriophage infection at the site of the bacterial infection in the target animal species. This is especially, the importance of this is in order to avoid unsuccessful clinical trials. And then also a representative in vivo model of infection might also be used. Then the immune response to the effect of bacteriophages should also be addressed. These data can come from the literature, and this is especially important in repeated treatment, and to document that this repeated treatment does not going to impact negatively in the therapeutic effect. And then for alternative bacteriophage combinations to the parental one, it may be possible to demonstrate efficacy based on the demonstration of comparability between preparations. For example, with a comparable biodistribution or a comparable immune clearance and MOI. Next slide, please. So then we have to take into consideration the possible development of phage resistance and related risk in animals. That is possible coevolution of bacteriophages and host bacteria, the risk of appearance and dissemination of resistant bacteria, the resistant mechanisms, and the molecular genetic basis of resistance. The peer-reviewed journals or proprietary study could be possible. And then it might be necessary to establish measures to limit the development of resistance in bacteria. Next slide, please. So regarding the dose determination and confirmation studies, the minimum effective dose, the proposed dose and interval, the duration of the treatment, and where relevant, any proposed repeated treatment should be provided for the representative monophage or multiphage preparation. This will be necessary in each target bacteria, each animal species, and the recommended route of administrations. The experimental models of infections in the target animal could be possible, and then a justification based on literature data may be considered acceptable, provided that the posology is supported in a preclinical or clinical study in the target animal species. And also these type of studies could also serve to evaluate the impact on the immunological function that I mentioned before. So regarding the tolerance in the animal species, it's necessary to characterize the safety profile before introducing in the field, that is the clinical trials. So this will be on the basis of the route of administration and dosage, included repeated administration and treatment duration intended for use of the product in its final formulation. A one-time dose is acceptable, and post-mortem examination should be omitted. And these studies are performed in healthy animals. However, we know that the safety data derived for the use of bacteriophages in diseased animals is more useful. That is that the safety data from the clinical trials is expected to be more informative. But whenever specific risks are identified, these studies could be required. For example, if the phages target also commensal bacteria. I think there was a question in the audience of how many animals are needed for these studies. I mean, this will be case by case, but we have also addressed it in this guideline. Other guidelines, for example, the BITS GL43 and BITS GL44, and for example, in the guideline of target animal safety for pharmaceutical products, they suggest like 8 animals per treatment. But I mean, these are guidelines that you can consult, and you can base your studies in these guidelines, or also other possibilities could be taken into considerations, just always case by case. So regarding the clinical trials, it's necessary to test under field conditions the efficacy and safety in the target animals. These clinical trials are in accordance with good clinical practice principles, and For example, these are points taken into consideration when doing a clinical trial, like the final formulation, including a representative preparation should be used, the diagnostic methods and clinical conditions of the animals. Study endpoints should support each indication, targeted bacteria and animal species. Also the clinical and microbiological inclusion and exclusion criteria. Isolation of the target pathogen and susceptibility in vitro test. The endpoints that could be the clinical cure rate or the microbiological cure rate, depending of the disease or what we want to obtain, and also the timing of the efficacy assessment, and the statistical methods could follow the guideline on statistical principles for clinical trials for veterinary medicinal products. But again, other designs could also be accepted. Next slide, please. So the claims that we can have or that we are expecting for these phages are the treatment, the metaphylaxis, and the prophylaxis of specific functions, the specific infectious diseases or infections caused by one or several specific bacterial species. We have taken into consideration special considerations for metaphylaxis claims. So metaphylaxis needs to be justified from an epidemiological point of view. The presence of the disease in the group should always be confirmed before, and then metaphylaxis is always in conjunction with a treatment claim, and the threshold for the initiation of the treatment should be justified on the epidemiological and clinical grounds, and a justification may be based on the literature. In the case of prophylaxis claims that we are talking about prevention, a fully justified for each target species and indication will be needed. Next slide, please. So then we have also taken into consideration this possible concomitant use with conventional antibiotics, just in case maybe laboratories want to study this. But any specific claim is required to be supported by data, by literature, and supported by studies. And then it could be considered if a significant therapeutic effect is demonstrated and if the risk of development of antibiotic or phage resistance are addressed. And next slide. Okay, so finally, we have include the post-marketing authorization changes that have already mentioned my colleagues before, because in the Annex 2 of the Regulation 6/2019, it is recognized that these products could be updated, as I already mentioned, because of the development of resistance or maybe changes in the epidemiology of bacterial pathogens. So it might be necessary to use a trained version of the phages already authorized or maybe new monophage components. So we have taken into consideration two different scenarios in two examples that we include in the guideline. For example, when we add a monophage which is comparable to one phage already authorized in the marketing authorization application. So this case will be very simple, or will be more simple, and we expect it's expected to be approvable based only on quality data. But if we add the second scenario is if we add one or more new components that are not comparable to what is already authorized, that then is a more complex example, and data from laboratory efficacy studies in the target animal species or representative species might be required, and in worst case scenario, clinical trials will be required. We also have to think that also alternative tools can be proposed, for example, surrogate efficacy endpoints that can be established and validated for the parental product, and this type of alternative use might be used. And I think this is everything. Thank you very much. And if you have any comment or any question.
Yes, there is a question in the room. Please go ahead.
Thank you very much for this this very enlightening presentation. I have a specific question when it comes to the endpoints of cure, of measuring efficacy. Yeah, as both the clinical and the bacteriological endpoints. Of course, we know them for several indications when it comes to antibiotics. In which way you think the efficacy, the assessment of efficacy of phage therapy will be the same compared to anti-infectives, or where do you think the differences will be?
I don't really know, but I guess, I mean, these recommendations of the clinical trials, actually they have been taken from the guideline of antibiotics. So I guess that the phages maybe are not as, has not the effect of the phages are weaker, let's say, than antibiotics. So I mean, it's not necessary to have a microbiology cure rate. It's depending on the disease that you want to treat. And I don't know if maybe Jean-Paul or someone there has a more clear answer, but.
So yeah, I don't think there's much recent data on the efficacy of phages. But from what we know, I can only advise from what I know from clinical practice in humans, is that we would never advise to use phages without combination with other anti-infective agents, such as antibiotics. We see mostly the synergy of phages with antibiotics as an important factor.
Thank you very much for that answer. So that answers a lot. So this discloses a lot. But then my question comes, how would you define synergy?
Personally, I said synergy, but an additive effect would also be nice. And there are definitions for that. If you look in the literature, there are definitions for additive effect or synergistic effect. Additive is like 1 + 1 is 2, and synergy is 1 + 1 is 10.
Yeah,
for
okay,
instance,
but when it comes to timing of the efficacy, I think that's a crucial one when it comes to phages. We know phages work very quickly, but also the effect it's different than antibiotics. Yes. And I think in that assessment, I think the timing versus whether you want a complete bacteriological negative sample, I think that's my personal opinion. Well, I think that would be very challenging for the approval of phages.
Yeah, I think it's very complex, and I can only speak from experience in human therapy. That is that we can sometimes see positive outcomes without eradication of the bacteria. And then when you look more deep into these patients, you see, for instance, that the bacterium became resistant to the phage, but became sensitive to the antibiotics, or becoming resistant became less virulent, for instance. So that's why I think, like, also this comment about like phages targeting LPS or efflux pumps. Personally, I would be in favor of phages like that, because we know that resistance will come even during treatment. But if this resistance then brings some other positive effects, such as synergy or making your antibiotic sensitive better again, or your bacterium less virulent, that would be something that you could see as a plus.
Absolutely. And then my question is, how do you translate that into the assessment of efficacy? Because that, I think, is one of the fundamental things when it comes to these treatments we talk about. Yeah.
I think it's also a problem in human clinical trials, because I think everything that we all the frameworks that we have today are made for defined drugs like antibiotics, and does not take into account agents such as phages. Huh? So I think we need to probably to learn. I think, yeah.
Thank you very much.
Yes, please go ahead on my left. Yeah.
I would like to repeat my question from the morning. If it's possible to change active ingredients throughout the trials, if you have like a larger collection of phages, how fixed is the combination when you go through the trials?
What I can say is that the guideline what it says is that you can choose like a representative preparation that you need to justify why you are choosing that a representative preparation, and then you use that for the different trials. You don't need to do trials with all different combinations, or I mean, you will have to justify why you are using that one as a representative one, and then use them in the different studies. I don't know if that answers your question, or if someone wants to add more things to the answer.
Yeah. So I think you mean changing your composition during the trial, that you find out, okay, in this animal, we would like to use another combination than in that animal. So I think that would make sense, but I don't know and how far this is dealt with by the guideline.
So I think what you just said, Belen, I mean, addresses that question. If during trials or at any stage during product development, you add a phage to your product or to what you intend to apply for in the dossier, then if that phage is still within what would be considered representative for the phages that you have already examined, then that should not cause any problems. But if you add a phage which is not, so to speak, outside of the representative space that you have established, then you may need to generate more data, so to speak, to encompass that phage. Does that make sense?
Yeah, I understand, but I think what Jean-Paul Penet was saying is a bit different, and that was more my question, that if you run into resistance throughout the trial, and you already have, let's say, 20 strains in your dossier, but you only used 10 for the trial, can you switch this within the trial? Can you switch from one phage out of this 10 against someone that another one that you did not use before, like from one animal to the next, or maybe even while you treat the same animal? That is it possible to switch within the phages you included in your dossier?
So in fact, what you would be evaluating in the trial would be your bank, and not like one combination. So you would do your trial with your bank, from which you would select phages according to what is needed in that particular animal. So the trial product would be the bank. Is that possible?
Yes. So I mean, the situation you outline is really about changing a clinical trial during the progress of that trial, as regards to the treatment in this case, or the factors, and that is always considered a complex issue. But again, if you do it the right way, then you will end up with having, so let's say you started the trial with 5 phages, and then during the trial you end up adding 5 more phages, so you now have 10 phages. So if you can, so to speak, justify that within the trial setting, so that the data that comes out of that trial are still valid, then you have basically shown that your 10 phages are efficacious, hopefully, in that particular trial, in that particular setting. and then there shouldn't really be any problem. But the problem always arises, I think, when you want to change a clinical trial during the progress of that trial, regardless of which type of product you're working with. And so again, in that situation, I think not only for phages, but really for any type of product, the advice would be to consult with the agency, because there may be special considerations as regards which methods you then need to use to analyze the results and so forth and so on.
I think it's very difficult to change your product during the trial, but if you would design your trial where your product would be any combination within your bank, like your bank is your product, and during the trial you just choose the adequate combination according to your animal, then what would be on trial would be your bank, and then you don't have to change your clinical trial.
Yeah, I think that what they are. I agree that the important thing is that when you design your protocol, you take into account that maybe it might happen something, and if it happens this thing, you will do. I mean, everything has to be established in advance. But I think it's the same as Martin have already said.
I think that at the end, your recommendation in the SPC would have to reflect how you have conducted your clinical trial and how the product should be used in the field.
Okay, thank you for that. And I think with respect to this issue, again, also that to define the endpoint very well is also important here. There has been a question. Dr. Kakabatse has raised her hand. Please go ahead.
Yeah, hello. Can you hear me?
We can hear you. Please ask your question.
So it's continuing the same topic about the representative formulation that goes in a clinical trial. I mean, there's a very high chance we will need to substitute the 5-phage, for example, that we have with another 5-phage. So, for example, the amount of phages in cocktail will not change, and maybe there will also be all myovirides will be switched with myovirides. Is this possible? And another question is if it's possible to do adaptation during the process also. For example, with the same animal, resistance occurred, and all the phages in dossiers show no effect, and then we are forced to adapt it to this resistant strain in an already treated animal in a halfway or something along this way. So is this will be possible, or it's not covered in this framework? Yeah.
I don't know. I didn't listen well to the question.
But I can start. I can start perhaps with the last part of the question. So my understanding is that the question was that you have a product and then let's say a product with flexible composition, and then it turns out that this flexible composition actually is not adequate, and you still have resistance, which is not covered by this flexible composition. So if I understood that question correctly, then you would be in a situation where you would need to do a post-marketing update to the product according to the guidance that we just discussed during the meeting.
Not post-marketing during the clinical trial. While
no, so then I misunderstood this. So
while trial.
If it's during the trial, then your question is very similar to the question that was asked just before.
Yeah.
So I think we have answered that, so to speak, already, that if I were to summarize, then I think the answer would be that yes, this could potentially be done, but obviously changes during clinical trials are always difficult. You should consult with the agency if possible at all. And in any case, as Frederick reminded us, whatever the outcome of the trial would be reflected then in the wording in the summary of the product characteristics for your product as regards indication and efficacy and so forth. Thanks.
I would like to add, if possible, you refer to adaptation of the bacteriophages to the patient. I think you are referring to training the phages, or you mean to adapt the composition, the flexible composition to that patient. What do you mean? Do you mean to use the phages that you plan to authorize at the end, or do you mean to train the phages to try to overcome the resistance?
So I'm using phages which are like this representative complex of the preparation. And I would need to adapt one of these phages because all of them, for example, lost their activity on a strain which evolved during the treatment that may happen. So what's consequence in the situation? Is it possible? So is this the same phage that I have in dossier, but it needs adaptation during the trial?
Yeah, well, I will say that this will be considered a deviation from the protocol because you are not using the product that you plan to use. So as Martin said, this will be consulted before doing that. And I will say that it will be difficult to analyze as results of the clinical trial. I think it will be difficult to assess the efficacy if you have this situation. So I will say that, of course, and as we said along the entire meeting, we should analyze the situation case by case. But I will say that moving more widely than what is described in the guideline, that is to allow flexible composition of the bacteriophages. I think that moving along that or beyond that, it will be difficult to analyze and to obtain a conclusion on that. So I think I will say no. You cannot adapt the phages during the clinical trial, but of course you can change one phages by another, as well as already stated in the guideline. Thank you.
Thank you.
I would say yes. If your clinical trial and your protocol foresee that this adaptation could be part of the manufacturing process, but that have to be done upstream of the clinical trial on the device of the clinical trial, and at the end, that needs to be qualified in the manufacturing process. And I think that could be done in the scope of the ICH Q9, 11, and 8. And I will have a short presentation at the end of this meeting around this issue. Thank you.
Oh, okay. Yes, there's another question in the room. Please go ahead.
Yeah, I think I understand that not everything is ready yet. When I see the efficacy evaluation, and I come back to that, I see it's rooted upon what is done with antibiotics. When I follow the discussion, I think there's room to see phages as a conjunct therapy, have you also looked at other regulations, for instance, like anti-inflammatories, which is a different way of assessing efficacy than antibiotics have? Have you also looked at that in order to apply that type of efficacy parameters to phages?
I don't think so, but it could be. It could be possible, and I think we are open to different approaches, so it might be possible. I didn't think about that before, but
okay.
All the guidelines could be applicable if it's scientifically justified. I think so.
And in that case, you would not you would register phage therapy, but you would register it as a conjoint therapy. Yeah? I don't know if I used the correct word, but a therapy next to other therapies like antibiotics or vaccines.
Out of curiosity, can you give an example of what sort of outcomes you are thinking of?
Yeah, well, for instance, increased clinical cure, for instance, quicker cure, like with an anti-inflammatory in this case. It doesn't kill the bacteria, the anti-inflammatory, but it does, in combination with an antibiotic, improves the let's say the disease process from the perspective of the patient. And I can imagine that there are phages, exactly how Mr. Pinet described them, who do next to attacking the pathogen also have other benefits for the patients. And if that only would be caught in bacterial cure, bacteriological cure, you don't catch that benefit. Yeah. So, and yeah, I think listening to this discussion here, I think that was something I would like to put on the table. Yeah.
Okay, thank you for that. I think there was another question, or no?
Perhaps can I just add a comment to this? So, just to remind us that it's actually stated in the new regulation that phage products should be based on lytic phages, and that is explicitly stated as far as I remember. So phage products, what is in scope for our guideline are lytic phages. That doesn't mean, of course, that if your product also has anti-inflammatory effects, such as Jean-Paul mentioned, that you could show that in a clinical trial and register that as part of the indication. But the phages in the product have to be lytic. And then, of course, any other effects they may also have, such as, for example, increasing susceptibility to antibiotics, which is already mentioned in the guideline, or anti-inflammatory effects or whatever. I don't think our guideline in any way stops you from pursuing such indications, but obviously you would need to show it in a clinical trial, and then it would be reflected again in the summary of product characteristics that your product can only be used together with such and such an antibiotic, and the only effect which can be expected is, let's say, that you get well 2 days quicker than you would otherwise have gotten without the phage. If that clarifies. Thanks.
I don't know if I can add something. So the thing is that also lytic phages can have other effects than the eradication of the bacterium. So the example was that a bacterium becomes resistant to the lytic phage, but it has a cost. It becomes less virulent, so there are less symptoms in the patient, less pain, for instance. So that's why also we look at the clinical improvement as an outcome. So, but I think that this can be done also in this case.
If I may, I only want to add that, well, the purpose of the guideline on efficacy was more focused, not in what guidelines to apply or what endpoints, but I think it's focused on allow a flexible composition of the product, that it was, I think it was preventing the development of this kind of products. So in general, the guideline have two main points. The one is that a flexible composition is possible, and the other is that you don't need to try every single combination that you want to use or to include in your product. So the efficacy part was drafted with this purpose. The applicability of other guidelines or approach, like you mentioned, the one of anti-inflammatory is of course valid, and we think we will go like this with this kind of products, because maybe they alone are not so good, but it was also considered in the guideline their synergistic use with antibiotics. So your comment is really, really very welcome, but I would like to point out that the purpose of the guideline is not to prevent the use of flexible composition, and all the parts were drafted with that purpose, that you can use flexible final products. Yeah, thank you.
Thank you for that. There was another question in the room. Please go ahead.
I have two questions regarding to livestock. The first, can we define endpoints using post-mortem examinations for efficacy if the condition is subclinical, zoonotic or stuff, or we need to apply under the EPSA scope? And the second one is, if for metaphylaxis claims, can we use the diagnostics of the previous generation in the same room, or a different pen in the same farm, for example? That's
Belen, would you like to say something to this?
The first question about the postmortem, I don't think I have an answer for that. I am not sure. For the metaphylaxis, please could you repeat the question? Is that I can hear you well? That if you can use the can you repeat it, please?
Yes, sure. For metaphylaxis claim, you showed that we need to prove the presence of the bacteria within the group. So I was asking if we can use evidence from the previous generation in the same animal facility, or from a different pen in the same farm.
But the thing is that when you do metaphylaxis, you are doing treatment and metaphylaxis at the same time. So that is that there are animals that are already have the disease, but you are going to treat all the animals because the animals in contact with these previous ones could be also subclinical infected or whatever. So you will have the disease in the farm at that moment, and you should. What I think is that you should prove that the infection is there in the moment of the clinical trial. You cannot. I mean, you have an idea of the disease because maybe you have previously had the same disease in the farm, but actually you need to identify the pathogens in the time that you are performing the clinical trial. I don't know if I answered to your question, but because metaphylaxis is not the same as prophylaxis, so the pathogen will be actually there in the time of the clinical trial.
Okay, thanks.
Yes, and with respect to your other question, I don't know if anyone else has any comments. I would think that in livestock there is use of post-mortem data for evidence of efficacy, like lung lesion scoring or gut scoring in poultry or whatever. So there's certainly a possibility. I guess we would normally look for at least a clinical advantage, but I think that that the data could definitely be an important part of evidence of efficacy. Yes.
There's one question in the chat again. So, yes. Could phages be regarded as agents against biofilm, not as antibiotics eventually? And would this make the process easier?
I can start perhaps here again. So I think use against biofilm is actually mentioned in the guidelines as something where phages potentially could have advantages even over antibiotics theoretically. I don't think it will make registration easier, but really the same rules apply that you have to show during your preclinical as well as clinical studies that your product has efficacy for the intended indication. So let's say it could be a biofilm in a orthopedic device or an endocarditis or whatever. You simply need to show that your product has efficacy in that setting. And biofilms, biologically, that setting, at least to my mind, would be slightly more complex than a setting where you have the bacteria, the pathogen in a more planktonic state. So I don't think it will actually make that type of products easier to develop. Thanks.
If there are no other questions at this point, then I would like to thank you for this discussion. And then we will, for the moment, leave the guideline and move to the perspective of the EDQM. And I would like to ask Olga Kolach to enlighten us on the activities with respect to introducing bacteriophages into the European Pharmacopoeia.
Thank you. Yeah, you can move one more click, please. Yeah, so I am Ola Colay-Robin, and I am a scientific program manager in the European Pharmacopoeia department in EDQM. And in this presentation, I'm gonna show you or explain the activities of the European Pharmacopoeia in the field of bacteriophages. Next click, please. Yes. So, but before let me please shortly introduce the European Pharmacopoeia. So it constitutes one common and compulsory standard and complemented by national pharmacopoeia. European Pharmacopoeia is the official pharmacopoeia in Europe, and it is applied by all the licensing authorities. As you know, it is legally binding and comes into force at the same date for all the members, while it is also used in hundreds of countries over the world. There are currently 40 members that signed a convention on the elaboration of the European Pharmacopoeia, 39 member states, with Albania being the most recent one from 2020, and the European Union. There are also 31 observers, I don't know, with 5 European, 24 non-European countries, with Ethiopia being the most recent country that has been granted the observer status, as well as Taiwan, FDA, and WHO. To give you some numbers, the recent European Pharmacopoeia Supplement 11.2 contained almost 2,500 of monographs, almost 400 general texts, and over 2,800 reagents. To talk about European Pharmacopoeia text, it is crucial to understand the structure of European Pharmacopoeia and different concepts behind the different kinds of texts. So they are divided into general notices, general chapters, and general individual monographs. I have someone speaking.
Could everyone, even those that don't think it's them, check whether they are muted? Thank you. Okay, it's muted. Thanks. Go ahead.
So, as I said, general notices, general chapters, and then general and individual monographs with the reference standards to support all those texts. So, general notices constitute absolutely essential reading before using the European Pharmacopoeia, and they address general issues, provide basic information, rules, and conventional expressions, and they apply to all the text in the European Pharmacopoeia. Then general chapters, on the other hand, are not compulsory per se, but they gain the mandatory status and become mandatory whenever they are referred to in a monograph. They provide standard analytical procedures and guidance texts, and they can be also used in case there is no monograph. Monographs, on the other hand, either general or individual, are mandatory. General monographs are overarching texts and describe quality requirements that are common for classes of substances, for example, monograph substances for pharmaceutical use, and dosage forms, for example, parenteral preparations. These monographs are mandatory for all the substances or preparations within the defined scope, and they are usually not cross-referenced in individual monographs, although, as you can see on the slide, there are some exceptions in the medicinal product monographs. And finally, individual monographs, they are mostly substance-based and specific, but they are not to be considered on their own. Many requirements are included in general monographs, which, as I just said, are not cross-referenced in the individual monographs, and in general chapters that become part of the standard when mentioned in monograph. Also, another feature of individual monograph that I'd like to particularly emphasize on is that they are elaborated and revised, taking into account products currently approved in Europe, and in the vast majority contain validated analytical procedures. So, as I also said, the European Pharmacopoeia text prescribe use of reference standards that are established specifically and exclusively for use described in those texts, as I outlined the chapter reference standards 512. So if you could click, please. So the European Pharmacopoeia texts are created and revised and maintained by a family of over 1,000 scientists divided into a group of experts and working parties. And the bacteriophage working party of the European Pharmacopoeia is among those most recently created, and it was created 3 years ago, with some discussion on the topic dating prior to this. So after nomination of experts and initial discussion in the group, the general chapter Phage Therapy, Active Substances and Medicinal Products for Human and Veterinary Use was added to the work program a year later, so in June 2021. The chapter was then drafted and was published for consultations last month in PharmRopa 35.2. And it is now open for comments, so similarly to the guideline that we were discussing earlier on. The comments are accepted. The public deadline is on the 30th of June, and with NPAs, with National Pharmacopoeia authorities, having afterwards 2 more months to comment. So I can only encourage everyone to do so. Click, please, yes. So now I wanted to show on this slide who Bacteriophage Working Party was. So it currently counts 16 experts from 10 different countries, mostly European, and half of the experts come from the regulatory side, both from human and veterinary fields. The other half represents hospitals and experimental institutes, as well as academia, and we recently have also a member from industry who joined. Next slide, please. The work of the Bacteriophage Working Party is quite high on the agenda of the European Pharmacopoeia Commission, and setting up standards for the quality of phage therapy is one of its priorities for the next 3 years. in the field of biologicals, next to such activities in like gene therapy or mRNA vaccines. Yes, now, yeah. So now if we look at the draft chapter 531 itself, firstly, it's a general chapter. So as I just said in description of different texts in European Pharmacopoeia, it is not mandatory per se. And since it is not referred to any monograph, it is published for information. It provides a framework of requirements for phage therapy products, their production and control. However, alternative approaches are allowed, subject, of course, to the approval by the competent authority. The setup is similar to the approach previously taken for gene therapy products. At the time of the chapter on gene therapy that was created around 20 years ago, no gene therapy products were approved in Europe. And the European Pharmacopoeia chapter provided a common framework, has been widely used ever since by all the stakeholders. And we hope that this chapter on bacteriophages could be equally useful and could do the same job, basically. The scope of the chapter is defined to be applicable to preparations of naturally occurring as well as genetically modified single phages or their mixtures that can be administered by various routes. So following definition section that I just briefly described, the main body of this relatively short chapter lists general provisions for production of phage therapy medicinal products, gives requirements for bacterial cell banks and phages used for production, as well as requirements for purified harvest and final lot of phage therapy medicinal products. The chapter contains also a short paragraph with considerations and requirements for adapted products, as well as a statement on labeling referring to relevant supranational and national regulations. So now going a bit more maybe in details. So according to the requirements of the chapter, the strain source for the bacterial master cell bank and its subsequent manipulation and tests used for its characterization have to be documented. Avoidance of strains encoding prophage, antibiotic resistance gene, toxins, and other detrimental factors is preferable. We discussed it and came back on it. As you can see here, it is written unless otherwise justified and authorized. The absence, if we go back one more thing, so master cell banks have to be identified by phenotypic and genotypic methods, including determination of antibiotic susceptibility profile and preferably whole genome sequencing. The absence of microbial contamination and the absence of detrimental phages should be confirmed, the cell viability determined, and the strain susceptibility to the phage demonstrated. So I'm not really sure if you click one more time, please, because no, so that the animation section, come back, please. The animations is not, I don't know why, happening, but you had in red requirements for bacterial, for working cell banks. So basically, the working cell banks are requested to comply with the requirements for microbial purity, cell viability, and strain susceptibility to the phage. Normally, they should appear in red. I'm not really sure why. Maybe you're in PDF or something. And also, you might have noticed some wording that it's employed in the chapter, being rather flexible, often or referring to other suitable method, if an example method is given for a test. And it's probably useful to remind that the term suitable is defined in general notices as to the satisfaction of the competent authority. And similar, the wording unless otherwise justified and authorized is also explained in general notices. And it means that the requirement is to be met, but the competent authority can authorize the modification or exemption in justified cases. So now please click one more time. Yes, so for phages used in production, the source of the master phage bank, its subsequent manipulations and susceptible bacterial species to the phage have to be documented. And similarly to the requirements for bacterial cell banks, the avoidance of phages encoding known or potential detrimental factors is recommended. again, unless otherwise justified and authorized. So this is this flexibility Martin was also talking about, and how it was aligned between the two texts. The phages used in production are to be identified by genotypic and phenotypic methods, including plaque morphology, whole genome sequencing, and for modified phages, the modification have to be described and their effect characterized. The phage purity to obtain a single phage clone with one plaque morphotype and potency defined as infectious phage titer have to be determined. And then a sterility of the phage in accordance to the chapter 261 is also required. And here again, I suppose you don't see it when you click again, but yes, so the last three bullet points, so phage purity, potency, and sterility should appear in red, and these are the requirements that are outlined in the chapter as those applicable for the working phage bank. So, and some flexibility words were also marked. So in this slide, I gathered requirements for production that are listed under both under general provision for production, but also under the section production and purification. So the phage therapy medicinal products are to be produced using a seedlot system with the suitable host phage combination in a way to avoid contamination between phages and bacterial hosts. The production process is to yield the phage therapy medicinal product of consistent quality and stability, while appropriate in-process testing is implemented at relevant time points and the key intermediate stages. The chapter also requires raw materials to be of pharmaceutical grade, and in addition, it refers to the requirements of chapter 5 to 12 for raw materials of biological origin. And now the requirements for purified harvest and final lot. In each case, the identity is confirmed using relevant genotypic and phenotypic markers, but in the final lot, the identity of each phage needs to be verified in case of the multiphage products. And similarly, the potency, defined as infectious phage titer, is to be determined in both stages by plaque assay or other suitable method. But in case of the final lot, it has to be determined for each phage. And this is also an aspect that has been brought today by Martin and came up in questions. So then microbial examination, as described in chapter 2612, is performed for purified harvest and either sterility for sterile products or microbiological quality with recommendation of chapter 514 for non-sterile products is required for final lot. The purified harvest is to be additionally tested also for residual reagents and host cell impurities and contaminants, while in case of final lot, additional tests to be performed are those for appearance, relevant pyrogens, water content and pH, the last two for freeze-dried and liquid phage therapy medicinal products respectively. So at this point, I'd like to perhaps highlight that the test for pyrogenicity, which refers to a new general chapter 5113, it's a work in progress because this new chapter has been published in Pharmaeuropa 35.1 with the end of consultation period at the end of this month, and this chapter describes provides the guidance on the choice of the pyrogen testing approach. And this is a part also of a wider exercise that affected many European pharmacopoeia texts, and it aims at the complete removal of rabbit pyrogen test from the European pharmacopoeia. And finally, the last paragraph of the chapter mentions the process of phage adaptation by which the phage therapy medicinal products are directed to evolve in order to increase potency against the clinical isolate from an individual patient. It defines quality of the phage or mixture of phages for starting such an adaptation process as compliant with the requirements of a section 2.3 which is a section that I mentioned before for phages used in production of phage therapy medicinal products. And it also lists the tests applicable to final lot of an adapted product, being those for appearance, sterility, sterility or microbial quality, depending on the character of the product, and pyrogenicity, while the potency of an adapted product, yes, is to be determined against the clinical target. So, as mentioned already, now, please, you can click. The draft chapter on phage therapy, active substance and medicinal products for human and veterinary use is currently published in PharmEuropa with the public deadline at the end of June. And we really cannot emphasize enough how strongly we encourage you to provide a feedback. This is absolutely crucial for us to produce standards that would meet expectations of all the stakeholders. I mean, this is, I'm repeating what my colleagues said before for the guideline, but it's equally true for European Pharmacopoeia texts. The link is to the PharmEuropa inquiry is provided on the slide, and the access is for free. You just need to register, so please do so. At the bottom of the slide, I actually also put a part of the European Pharmacopoeia text life cycle from the PharmEuropa publication to give you an idea what might be the next steps. So after the public inquiry, all the comments are, of course, reviewed and the text is finalized. And it can happen that the text might need to be republished in PharmEuropa for additional round of inquiry, and that, of course, automatically adds additional 5 months to the process. But once the text is finalized, it is then submitted to the European Pharmacopoeia Commission for adoption. That, if happens by consensus, so everyone has to agree. And after this adoption, in this case, the chapter would be published 6 months later in a supplement of European Pharmacopoeia. And the supplement will come into force 6 months later, and additional 6 months. So basically, the supplement comes into force a year roughly after its adoption. And then I just have one last short communicate. If you do want to become a part of the European Pharmacopoeia family, and if you do want to actively help shaping the text of European Pharmacopoeia, just join the network. Thank you very much.
Well, thank you for this commercial break. And then thank you very much. Perhaps there are questions here. Yes, there is a question in the room. Please go.
Is there any difference in the threshold of endotoxins, impurities, or whatever it's there? Depending on the route of administration, is it going to be different for injectable to oral administration of phages?
So this aspect certainly won't be covered specifically in the chapter, but it does refer to the chapter on 5113, which is not yet a compendial chapter either. That will describe all the process, how to choose the best approach for pyrogen testing. And then you do have these aspects. I don't think there'll be now. I don't really remember whether there are actually numerical limits, but yes, it is known that you would have a different bacteria endotoxins limits if it is an injectable or if it's a different administration route. But this aspect won't be like, it is not included specifically in the chapter that we are talking about.
And I have another question. So it's about the weight you give to the presence of unwanted genes encoding antibiotic resistance and virulence factors that we discussed before as well. Here in your presentation, you said that, if I understood correctly, these are allowed also for phages if we can justify them. It's very surprising for me that the weight for a phage, the weight you put on the presence of these unwanted genes in the phage compared to the host is equal. For me, it's really, I don't understand this.
Yeah, I got this already from the previous discussion and the discussions here that Martin was replying to the questions. The idea is the same, and our both texts are aligned here. The idea is that we want both of them as clean as possible, but it doesn't mean that that certain things won't be allowed, right? Because it will depend on the situation itself. However, I see your point, and I would only encourage you to comment on the chapter, probably on the guideline as well, and then we will take it into consideration. We'll rediscuss it and potentially adapt the wording to make things clearer.
There is another question in the room. Oh, Paul, you want to say something? No? Yes?
Well, I don't want to add anything. I also had a question. So if the other person was first.
Okay, well, you go ahead, Jean-Paul.
So my question is why did you add mixtures and medicine products into this general chapter? Because for me, if you are making a mixture, you're going more for like the defined product, and you're more in what already, for instance, the guideline we are discussing here, or like the general framework for medicine products. Because I think like when you make an active substance, or you make many active substances according to this, maybe the future monographs that will be linked to this general chapter, then you have all the flexibility. Then you can just mix the phages that are active in the pharmacy, for instance, or on the farm, and then you have a sustainable product that only uses the active phages. So my question is why the mixtures and the medicine products?
Well, the scope of the chapter was the subject of a great discussion as well, and it ended up being both the substance and the product to go till the end, to provide the guidance, the framework till the end. Now, that's true that it does talk about the single phage product and the mixture of products, and it doesn't have all those aspects that the guideline just discussed, you know, how you mix them at the end and how flexible it can be. It defines the quality at the end. So if you do have a combination of phages in the product, you still have to meet several criteria that are listed.
Yes, I understand this, but personally, I would not see any advantage of making a mixture, because it will complicate my life, because I would have to determine the activity of each phage in the mixture, and the only activity that really counts is when you go to the patient or the animal, and then you select the phages that do work. So I don't see any advantage of adding mixtures to this chapter.
Well, it was to provide the framework, as I said, till the end, and also maybe what I could say is that the work of the bacteriophage working party, of course, it's still not done within this chapter either, because now we are in public consultation phase, and then we will have to review hopefully many comments that you are going to provide us with, and then finalize the chapter. But we are already thinking of what else could be of use, and this potency aspect that we already discussed today, and the questions that came, how to do that for each phage and so on. That was, of course, it's nothing new to the group. It was heavily discussed as well, and the group is currently proposing to draft a text on guidance how to tackle this potency. So this, of course, as I said, it's just a proposal, and the work might start only if it is approved by the European Pharmacopoeia Commission to be added to the work program, and then we might have a bit more into this potency. But I mean, the question to answer why you do products, because the chapter is for both, for substance and the product, so to come to the end. Yes, but I fully understand that it becomes more complicated if you want to do the mixtures.
Dr. Chartrand has raised her hand. Do you want to comment to this point, or do you have another question?
Yes, good afternoon, everybody. This is Emmanuelle Chartrand speaking, and I'm a colleague of Ola. And I just wanted to emphasize, indeed, so I have a comment to this questions that indeed there was in extensive discussions among the experts group of the European Pharmacopoeia, and what they call the cocktails, or these multi-phage products, are indeed administered. So there is some practice to do that, and it was to cover what is done. And there are experts around the table who do in fact use these mixtures of phages, and it is important that the European Pharmacopoeia chapter covers that. And as said by Ola, this is more complex, but at least some hints will be given on what to address, because indeed mixtures, as soon as you have mixtures, there can be problems of interaction or inhibition. And I just wanted to say that there was similar discussion in a monograph of the Pharmacopoeia, you may know it, which is the live biotherapeutic product monograph, which has some kind of similar type of problematics, and so to assay the potency of a mixture of such product is a challenge. And just to come back on the question on the bacterial endotoxin limits to be used, it is considered that there is no specific limit. There are single limits that are provided in the Pharmacopoeia in chapter 5.1.10, which provides K values on a value to be used to calculate the limit based on the route of administration. So bacteriophage would not be a specific case, so this general rule would have to be applied. So just a complementary information. Thank you.
Thank you very much for that. Then now we will go to my left. There's a question there.
Hello, Frank Smecker, Jofran. Two questions. One is you also mentioned to do the sterility test according to Pharmacopoeia method 2.6.1. So does that mean that for each phage, also the method for sterility for each phage needs to be verified, or that can be done for one phage and then you can do testing for any phage later? And then the second one, just so these products are then, that's a general question, released by the QA, or needs to be the QP as GMP, or the QA in the laboratory? Thank you.
Well, the last question, I don't think I don't know these specifics. I mean, normally it's European Pharmacopoeia describes the quality of medicines and the substances in it, right? So now it does take into consideration also unlicensed products, but that wouldn't be, again, phages wouldn't be anything different. It would be still the same, in the same framework. And then I forgot the first question.
The first one was just whether, for example, sterility method, which is Pharmacopoeia method, would that need to be done, verification of the method for each new phage separately, or you can do once? And that is actually the same for also for other QC methods, whether that needs to be once for any phage, or that needs to be for each phage separately, verification.
So you're referring to the final lot testing. So it depends what your final lot would be. I would imagine that if your final lot is defined as a mixture of phages, then you test the sterility on that. However, in terms of identification and potency, it is clearly stated in the text that you have to do it, demonstrate for every single phage.
Right, but for sterility, you can do it, or endotoxin, for example, right, which is more independent from the potency. You do the qualification of the method of endotoxin or sterility verification once, or you have to do for each phage that you are releasing each final product or cocktail?
For now, I think we are maybe not having the same terminology in mind, because the final lot, when you say final phage, what does it mean? One final lot, or one of the phages within the final lot?
Let's say you're producing phage A, right? And then you're doing the sterility testing, release testing, and in the GMP you have to do verification for that specific phage A product, and then you're producing phage B separate product, separate lot, would you need to do the verification of the sterility as per pharmacopoeia again, or
if your phage B is a separate product, yes.
I can add, if I may. The term you use are a bit ambiguous. So the sterility chapter 261 has a part called suitability of the method in the presence of the product, and you have to do that for every single phage. You cannot assume that if you've done it for once, then it will be the same for the others. So if you follow the method 261 step by step, then you have the answer. So the suitability has to be done for every single product.
Okay, thanks. Yeah, I meant suitability, but yeah, thanks.
Okay, is that I wanted to say, because I understood that you were talking about suitability. So.
Okay, Jean-Paul, you have a comment to this point?
Well, yeah, just as an additional comment. So, of course, yeah, it's common practice to use mixtures. We also do, but we rarely use the same mixture. We just choose the components of this mixture according to the bacterium that we want to target. But let's say that you would use a mixture. And let's say you want to make a mixture for veterinary use. Who will decide whether you would go EDQM or EMA? Because it seems then that you have like two defined products that are mixtures of phages, and that could go either way.
No, so the guideline applies to veterinary phage medicines. So if you want to register a phage medicine, regardless of whether it's a mixture or not, regardless of whether it's a monophage product or a multiphage product, on the veterinary side, because that's the only site where we have a guideline, if you want to register such products, you need to apply centrally to the EMA for a marketing authorization. And the EDQM text is, as I think also you mentioned, Ola, not a guidance, but rather some general hints or informations about methods which can be applied. I think we can say rather specifically for characterization of the phages. And the texts, I think, as I also mentioned, are aligned, but obviously should be read together. But again, the guideline is the guideline, and the pharmacopoeia text is separate.
But at this moment, it's not mandatory. But let's say in a few years there will be a monograph, then it becomes mandatory, and then you have like two different.
But again, it's not that situation exists for all products. So for all products, whether you talk about medicine, vaccines or antibiotics or whatever, on veterinary and on human sides, you have guidelines, EMA guidelines, and you have pharmacopoeian chapters, specific chapters or whatever, and those exist side by side.
It's a bit strange that for like the same product you could have done different guidelines.
But again, it's not a different guidelines. So this situation exists for all products already.
Sure.
And the guidelines, there's only one guideline, so to speak, and that's what EMA puts out. And then the pharmacopoeia text, correct me if I'm wrong, lives together with that, and you could say carries equal weight, of course. But those two texts are supplementary.
Yeah. Shouldn't it be better like just to work together on one text?
If I may, the scope of the EMA guidelines and the scope of the EDQM are different. EDQM, I will say that is wider. The scope of the EDQM is wider, is for human and for veterinary, and it's in general focused on quality, not for the vaccines, because they also address safety and efficacy, at least in veterinary. And the scope of the guidelines from EMA are for veterinary medicinal products as defined in the Regulation 6 2019. And I think that the key point here is the industrially process. So EDQM chapter applies for not only for industrially manufactured medicines, but also for magistral formula, as an example. But the guideline that we are discussing today apply for industrially manufactured, in this case, phages. So the scope is different. That's why we need to duplicate the work.
I understand, but I come back to the initial comment. Is for magister preparations, why would you make defined mixtures?
You mean in the EDQM?
Yes.
The mixtures?
Yes.
Well, because I think if I, and correct me that you are the EDQM expert, I think that EDQM chapters and monographs and so on try to regulate the product, not only the active substance, but also the final product. I think it's part of the template or working procedure in EMA.
Susanna, please,
Yes?
can I interrupt you?
Yes.
I think we've lost the connection to everyone. If anyone is still there, like Emmanuel, can you say something?
Yes, I follow everything. So
very good. You're still there, because we don't have anything on screen here anymore. But I saw that you raised your hand. So if Susanna has anything further to say, and then after that you can comment.
I finished. Thank you.
Oh, thank you. Well, Emmanuel, what do you have a comment to this?
Yes, I have a comment. First of all, sorry, I don't know who talks. So a gentleman says that why not work together? We are indeed working together, and Martin, who gave the second talk, Martin is indeed part of the expert group from the European Pharmacopoeia. So, and we are also observing the group from the EMA, and so we are working together. There is no contradiction or no conflict at all between what is the EMA guideline addressing and the European Pharmacopoeia text. So don't be worried about that. So that's one remark. And then a question from another gentleman saying, who should we go to if we manufacture such and such? You should not go to EDQM, because the EDQM is not an authority. The EDQM hosts the European Pharmacopoeia, which provides guidelines, quality requirements on medicinal products and their constituents. So if there is something described in the European Pharmacopoeia text, it does not mean it is approved. It is the authorities who are going to look at what they receive in their file, and they're going to look at the official text of the Pharmacopoeia and make decisions based on that. So the EDQM and the European Pharmacopoeia have no authority there. And then the fact that the European Pharmacopoeia describes something which is in fact a practice, why I mean, I don't know personally, but those around the table, they probably, they know why. So once again, these questions can be raised during the consultation. Thank you for giving me the opportunity to comment.
I've been trying to press the speaking button for quite some time. So thanks. I wanted to say actually mostly what Emmanuel just said, but to complement perhaps to say that to emphasize that the guidelines of EMA and the European Pharmacopoeia text are absolutely not contradictory. On the contrary, guidelines sometimes refer to the chapters of the European Pharmacopoeia, and we are of course following the guidelines, and in this particular case, we did have this link in the Martin as a link between the two groups. So there is a synergy between those, and also what I said during my talk, I would like to perhaps re-emphasize this, that this is the similar situations like we had for gene therapy products in the past, and the fact that the chapter was created some time ago and was viewed very helpful by the stakeholders on both sides, regulatory and industry sides, and we are hoping for the same. And then other question, this is the scope of the European Pharmacopoeia to come up with texts for medicinal products and their constituents, and this is what we are doing, and we are hopefully going to help stakeholders both on the industry and regulatory side to progress. And yes, the scopes are very different, but they are aligned in those where they overlap. Thank you.
Thank you very much for that. And then I think we will go for a short break of, what is it, 15 minutes, just a short bio break. So we will be back at 5 minutes past 3 o'clock. 3 o'clock, sorry. Yeah, 15:05. Okay, welcome back, everyone. There was a question in the chat on the access to the slides that have been presented, and these will be published hopefully within a few days on the event webpage. So they should be then available to everyone. The next part of the meeting is the presentation of some case studies. We have already had some good discussion on also practical application of the guidance, but these case studies might further illustrate how we think it should be practicable. I think I'm not sure who is going to present first. My colleague Susana Casado will present the first case studies. Please go ahead.
Thank you. I will start with this case study that is follow the guideline as it is drafted, and after me, then Frederick will present another case study illustrating the flexibility that we give to this kind of products. That's why I'm starting, and after that he will follow. So this case study is about a veterinary medicinal product with flexible composition to treat diarrhea caused by enterotoxigenic E. coli in post-weaning piglets. Next slide, please. It's important to say that this case is partly hypothetical, and examples including description of assays, parameters, or studies are not binding for applicants. It's only an example. And this case study is only intended to provide a high-level example of the principles described in the guideline. Next slide, please. A brief introduction of the target bacteria. The enterotoxigenic E. coli are pathogenic for some piglets at post-weaning when they express certain fimbriae and toxins proteins, and the most common pathotype is F4 STB-LT. Next slide, please. So here we have an example of a bacteriophage medicinal product that is planned to have flexible composition from a parental preparation that is composed by well-characterized natural phages that has been isolated from sewage in a pig farm. The indication of this medicinal product is the treatment of diarrhea caused by enterotoxigenic E. coli, and the posology and the route of administration is oral administration during 7 consecutive days after detecting this disease. The target species, as I already said, is post-weaning piglets. Next slide, please. So the first step is to obtain this product and try to register it is to isolate and characterize the bacteriophages. In this example, the phages were isolated in sewage in pig farms. There were 4 rounds of isolation, and there were 4 bacteriophages isolated. Is EC1, EC2, EC3, and EC4. So they were characterized, studying their morphology, their plague site. The plague stock was prepared, and the bacteriophages were also characterized in morphology by an electronic microscopy by an electron microscopy images. Next slide, please. The genome was also studied, searching or looking for toxin, virulence genes, lysogenic cassettes, or antibiotic-resistant genes. And in this fantastic sample, there were no any of these genes present in these phages. Their growth in vitro in a specific E. coli strain was also studied, and the growth on this strain was studied and compared between the different bacteriophages. And the most relevant part of this characterization was the study of the lytic activity of these phages in a representative battery of E. coli strains. In this example, the bacteriophages were tested in 89 bacteria strains, and as you can see in the graph, EC1 was able to lyse almost the 65% of these 89 strains, in contrast with EC3 that was only to lyse below 20% of the strain tested. Next slide, please. So based on this characterization, the initial candidates for this multiphage product were EC1, EC2, EC3, and EC4. However, the applicant or the developer removed EC3 from the equation because it didn't show a wide host range, and the growth properties were not as they expected. So the parental product was defined as EC1, EC2, EC4. So that means that for each treatment, a combination of these three bacteriophages could be used. But in order to avoid unlimited clinical trials and preclinical studies, then the developer select a representative preparation for the safety and efficacy studies. This representative preparation is well justified and should be well justified and tailor-made for the safety and or for the efficacy studies. Next slide, please. So the next question, after having the quality more or less control, is if my product is safe. For this, and based on the guideline, we based the studies that we need to ensure the safety on a risk profile analysis. It's allowed in the guideline to use scientific publications, previous experience, in vitro data, and of course in vivo studies, laboratory studies, and clinical trials. The safety part of the dossier or of the registration includes safety for the target species, safety for the user, and safety for the environment and for the consumer. So for the target species, this could be addressed in part 4, efficacy. And the typical study for safety is the TAS study, the tolerance in the target animal species study, that is a preclinical study. Well, when this hypothetical applicant apply the risk management system to determine if the product is safe or not, they identify that the commensal flora could be affected by these bacteriophages. And to address this potential safety problem, they design the TAS study in the efficacy part, as you can see below. So the objective of this study is to address the identified safety risk identified, so is to address if the commensal flora could be infected by these bacteriophages. The target species used in this hypothetical laboratory study is healthy piglets at post-weaning. The treatment is following the expected posology, and the study parameters are the comparison of the composition of the commensal flora before and after the treatment, and also between the treated group and the control group. The results for this study is that it was not detect a significantly disturbed of the commensal flora in the target species. Next slide, please. As I said, the safety package also includes the safety for the user, and as Sandra explained before, there are no specific guidelines for this kind of products, but the guideline you see below can be followed. And in order to determine the risk and to identify them and include in the SPC the warnings needed. In relation to the safety for the environment, and as also explained Sandra very well, a reflection on it should be included in the dossier. And for this specific case, it was concluded that no impact to soil bacteria and soil function can be identified using this product. And safety for the consumer, I think we already discussed about this a lot. EMA was consulted and it was decided that there was no need for MRL for this hypothetical product. Next slide, please. So now we move to the efficacy use of the product. We already know during development and during the characterization that the product is efficacious, at least in vitro, because we test the bacteriophages in a representative sample of strains, and we have more or less the host range established in vitro. However, we cannot derive from this in vitro information what the posology should be and if there is any interaction with the immune response. For that, we need to use our product in animals, in the target species, by laboratory studies and clinical trials. So, as I already said, for this we can use that we call in the guideline representative preparation to do these studies. And the information that we obtain from these studies is extrapolable to the other bacteriophages because they are comparable. And they should be comparable. This comparison should be based on representative and validated in vitro and in vivo data or parameters, and is up to the applicant to decide what these parameters will be. And also a comparable biodistribution, immune clearance, and multiplicity of infection is demonstrated. So if we can conclude that our bacteriophages that we use to test in the studies are representative for all the bank, then we can use all of them. And also it's important to include in the dossier a reflection upon the risk of developing and spreading resistance in the target animals. Next slide, please. So this is an example of a potential laboratory study for dose determination and dose confirmation. The medicinal product that we plan to administer or that we administer to the target animals is the representative preparation. In this example, we use different doses because we are not entirely sure what is the minimum effective dose. So here we treat post-weaning piglets, of course, and the group of treatment are control versus treated animals. The animals are challenged with a specific strain, the most frequent in the field, before treatment, and then the product is administered. And the parameters studied are the clinical signs related to the disease. There is also another parameters about the setting in phases of the pathogenic strain, and also a study of the immune response. The results and conclusion on this study are that the higher dose of the representative preparation was able to reduce the clinical signs and the setting of tested strain, and no immune response against the bacteriophages was detected. Next slide, please. So we end our development phase with a clinical trial. And here we again use the target species in which we administer the representative preparation previous slide, please. And the study parameters are the clinical signs of the disease and setting of the pathogenic strains. And the conclusions of this clinical trial are that the parental preparation shows it to be efficacious to treat the disease, and no safety concerns were identified along the study. Next slide, please. So we have finally our product. It's perfect, it's safe, it's efficacious, and it's ready to be marketed. So we prepare the SPC, and in the SPC, we reflect here the parental preparation, that are all the potential bacteriophages that we can use. But in the labeling, we should reflect the actual composition of the final product. So in the SPC, we will read the parental preparation, that it could be EC1, EC2, EC4. And for each product used in the market, it could be EC1, EC2, EC4, or EC1, EC2, or whatever. Next slide, please. So 2 years after this first release of our product, we identified that there is a bacterial resistance developed against EC2 in the field. So we as developer wish to update the marketed product with a trained version of the EC2. So we optimize these bacteriophages, and we want to include it as part of the parental preparation. So then we need to present a variation, and the requirements applicable to this new version of the phage EC2 will depend on the comparability of this new version against the original one. Next slide, please. This part is summarized in the annexes, and I think that my colleagues explain it very, very well in the previous talks. And in general, if the new version is comparable to the original one, then the requirements will be restricted to quality data. But if it's not comparable, then maybe it's needed to do new safety and efficacy studies. And next slide, please. So we guess that our new trained bacteriophage is registered. Then we need to update the SPC and include this new version of the bacteriophages in the SPC. But the labeling again should reflect the actual bacteriophages that are part of this specific final product. Next slide, please. So I include here some take-home message and conclusion from this case study. And these are that the characterization phase is really important in order to have the product well controlled and to prevent studies that don't give us any conclusion. It's important to obtain data to support the comparability of the bacteriophages, to support the flexible composition of the product. The risk profiling methodology to identify inherent risks to the specific product will help to decide the studies that we need to support the quality, safety, and efficacy of our product. Next slide, please. And product updates should be considered from the very beginning of the development of the parental product. This will facilitate the inclusion of new versions or new bacteriophages to our pool of bacteriophages, and the elaboration of plans and protocol for anticipated product updates formalized in post-approval change management protocol will foster the authorization of this new version and of these new variations. And next, and yeah, that's all from my side. Thank you.
I think we go straight on to the next speaker for another example. Is it right? Okay, Frederick.
Thank you, Jacqueline, and good afternoon, everybody. I will try to be brief to respect the schedule. Next slide. I have the same disclaimer as Susanna. The following example has only been provided for illustration of this draft guideline, and especially the flexibility that could be accepted for a product taking into account ICH Q8, Q9, and Q11 guideline about manufacturing process. This example is purely hypothetical and does not rely on a publication. And the objective of this presentation is just to spark discussion on the guideline. Next slide. And next slide. This new case is about a variable multiphage preparation of 3 phages, and this product is dropped over 3 days into the ear of dogs for the treatment of external otitis caused by Pseudomonas aeruginosa. Next slide. The 3 phages are chosen from a bank of 8 phages, which have been comprehensively characterized, and their specificity has been challenged against Pseudomonas and non-Pseudomonas strain, especially other bacteria of the ear flora. The phages harbor no genes anticipated to have a detrimental effect on the safety and or the efficacy profile of the product, for example, toxin or resistance genes. Okay, sorry. 4 other phages were initially considered for inclusion into the bank, but they failed to pass all the criteria. Next slide. The 3 phages of a batch are chosen in accordance with the result of a fibrogram of the strain isolated from the ear of the dog to be treated. Therefore, there is no need to define a representative preparation to undertake clinical trials, and each treatment is carried out with a single batch. Next slide. The bacterial propagating strains are also qualified to consistently yield phages with consistent quality, and this propagating strain harbor no detrimental gene in the final bulk of the phage. Next slide. This slide details what should be fulfilled in the section qualitative and quantitative composition, and there is not much to say. Next slide. Each batch is manufactured according to GMP rules, which will soon be adapted to novel therapies by a dedicated EMA group, and in accordance with Regulation 2019/6. The 3 phages to blend in the final product need to have an efficiency of plating higher than 0.1 in the phagogram. Next slide. Otherwise, they can be trained with the isolate from the otitis to be treated. The risk associated with this training, for example, the potential transfer of bacterial antibiotic resistance gene, has been justified as negligible because the product will be administered to the animals which already harbor high amounts of this training strain. Next slide. For the safety package of each phage of the bank, this is similar requirements to the previous examples. So I will skip this part. Next slide. No specific toxicology study has to be submitted, and the corresponding risks for target animal, for the user, and for the environment are addressed with the same kind of data as those included in the pharmacology section. Of note, the necessary data are limited because this is an external treatment administered to a non-producing animal. Next slide. This is the same remark as Susanna did. So next slide. For the efficacy package, pharmacology data for efficacy are addressed the same as for safety. Reflection of the risk of development and spreading of phage resistance is provided. Next slide. An experimental study which determines the dose regimen of the product is conducted. Otitis was triggered by dropping Pseudomonas aeruginosa strains into both ears of laboratory dogs. One ear was treated by the product and the other by saline, and the clinical and bacterial cure was monitored. There is no specific confirmation study in the efficacy package. The pivotal clinical trial would confirm or not the efficacy of the treatment. Next slide. A tolerance study was performed in few healthy dogs where their air flora was monitored before and after treatment with the batches used for the dose determination study. Next slide. Finally, a pivotal clinical trial was performed with veterinary practices. Dogs with external otitis where Pseudomonas aeruginosa was isolated were blindly treated either with a product or with saline. Again, the clinical and bacterial cures were monitored, and the result of the experimental laboratory study were corroborated. Next slide. Next slide. Thank you. And finally, you see the summary of product characteristics, where all the phages are listed with the information that 3 phages only are included in each product. On the label of each batch, the 3 phages are mentioned, with the information whether they are trained or not. Next slide. Finally, a risk management plan, which is mandatory for novel therapy products, enables to get systematic pharmacovigilance data about efficacy and safety. Next slide. When no phages will met the 0.1 efficiency of plating after training, the phage bank should be updated with new phages. This so-called 4Ks have been illustrated by Susanna, and there is not much to say, so I will stop here. Thank you.
Thank you for that, Frederick. So my question to the audience is whether there are any questions specifically on these examples that are given now, or also, I think, other general questions, since we have arrived at the finalizing discussion of the day. So if you have any other burning questions, then please go ahead. Yes, please.
Thank you very much for Susanna. I have a question on the safety assessment you gave. I know it's a hypothetical example, but on the safety assessment, you judge safety on modification of the gut flora. And I assume, well, that when the gut flora was not modified, you considered it as safe. But if it's modified, then the question is there a safety aspect? Because I would assume that safety would be reflected in, for instance, lesions or something else, or clinical symptoms. Yeah. And so basically my question is, how relevant is modification of gut flora for safety assessment?
Thank you for your question. Well, as you said, this is an example. I will say that if we see with another medicines like antibiotics that if the flora, the natural flora is altered, then there will be adverse events like diarrhea after having antibiotics. So in this case, I think that is if the flora of the animals it's affected and is altered, then maybe we can see some adverse event during the study. And that's what was the study was focused on. So maybe another potential parameter that I don't include in the case study will be if there were clinical symptoms of the variation of the flora. Now speaking with you, I think it will be a good endpoint for this specific case. And I think it's relevant to study if the product is specific enough. And if you identify that your bacteriophages could alter the non-target flora, I think this is relevant in general. Yeah, but maybe a better endpoint will be if there are consequences of these alterations. Yeah, thank you.
Thank you for that. Are there any further questions at the moment? Although there were also some questions in the chat, I think. Oh, is there any hand raised?
Yes, there's a hand raised. Eline Kakabache.
Mhm. Yes, so as I understand, you can perform a phagogram on the isolates of the participant animals beforehand and adapt the phages and go through the trial, but no, don't do any changes meanwhile. It was,
The question was, I think, that you can apply the phages according to a phagogram that was tested, before on the diseased animal, but not change the phages applied during the treatment. I think that was the case, in this example, but that was the question.
I'm not sure if you are referring to my case study or to Frederick. I think it's the Frederick ones.
Could you repeat, Jacqueline? I have not catch the question.
Yeah, so I think it was understood that the dogs were treated according to the results of a phagogram that was prepared before treatment. Yes, that is correct. Yes, that is correct. Yes, but then the question was whether the phages used were then not changed during the treatment. There's a similar question as before.
But it's not a question as long as the treatment is efficacious. And normally the phage are supposed to be lytic. So what is the goal is first of all the clinical cure and hopefully the bacterial cure, and if we don't expect that there is no resistance to a cure, but this resistance will be not detrimental to the treatment. Maybe I have not responded to your question.
Yeah, sorry, my internet broke meanwhile. Another comment is about the 3-phage preparation. I think this is quite optimistic. I would say that the initial bank probably should be around 100, and then the flexible amount of dossier phage is 30, and then from this amount of working phages should be about 7 to 10. And in this case, the production costs go very up. And this is a concern that it will be not feasible to formulate this type of preparation that will show efficacy, because we saw many times in human trials also that with this low amount of phage mixtures, it usually fails on efficacy level.
Yes, as I have told at the beginning of my speech, it's a purely hypothetical case, and what I wanted to emphasize that the training of a phage can be part of the manufacturing process and not a post-authorization modification. And maybe I should have include more phages on a more complex design, but yes, it was, and for this kind of bacteria. But again, it's just purely hypothetical to emphasize the flexibility of the approach on the guideline.
And I also think that in general, this problem, as you say, was understood by the group and it was definitely discussed, but we have to work within the framework of the directive or the regulation. I mean, so yeah, there are limitations to what is possible, but I think the guideline was designed taking into account trying to give as much space, so to say, as possible, and taking into account the issues with the production cost also. I don't know, Martin, you're nodding, whether you want to comment on that.
Perhaps just not so much to the manufacturing costs, but I think one thing that was mentioned in the question was also the formulation challenges, and you know, for a product containing even many phages, you don't necessarily, obviously, if you feel that it's not viable, have to pre-formulate it as a mixture. You can, that's up to you, formulate it as single phages or as less complex components, which can then be mixed at the farm, for example. That's completely up to developers and can perhaps help address some of these challenges. Thanks.
Thank you for that, Martin. Were there any further points?
Yes, there are several questions in the chat. So the first one is, during the EHEC epidemic, we learned that phages have been used for packaged food since long. Can we draw some information from the respective guideline on phages used on food eventually?
So we have actually, during the drafting of the guideline, looked at, I think that was also mentioned during Susanna's initial presentation, for example, the GRAS listing, generally recognized as safe listing of phages in the US, where some of the approval documents are publicly available, and also these two products that Susanna mentioned that have been approved in the EU under the as feed additives under EFSA auspices. So we have looked at that documentation, and I think to the extent where there are overlaps, for example, in terms of how phages should be characterized, you will find that what we have written in our guideline would probably align very well with considerations in those other situations. But obviously the scopes are different, so it's a different set of regulation which applies to feed additives and to medicinal products. So I think we have tried to learn from the existing products, but obviously also we have had to draft our own text. Thanks.
And if I may add to that, we had observers from both the FDA and the EFSA, so there was definitely contact and at the scientific level discussion with those two parties. So yeah, we have taken it into account is the answer.
So the next question was raised, I think, regarding Susanna's study. Would such a study really be approved by an animal welfare body and ethics commission?
I think I don't catch the question.
The question is whether the study you presented, the hypothetical study, I must say,
yes,
whether that would be feasible, that it would be approved by an animal experimentation committee or an ethical committee. To be honest, my answer would be yes. I wouldn't know why not.
Maybe that's what
this type of study.
I would like to say the same to you.
Yeah, maybe unless you propose to use 500 pigs, but otherwise it should be possible.
Yeah, I think it's more or less standard in the pharmaceutical development to control group, treated group, and so I don't see the problem with the design that we propose here. But thank you for the question. I hope so. I hope it will be welfare in accordance. Thank you.
And also regarding your study, can you share the protocol of the study?
I would like to, but it's confidential, you know. No, there is no protocol here. It's based on the experience that all the group have on assessing another dossiers, especially biological medicinal products. And well, we agreed about the endpoints as an example, but as we said at the beginning of the case study, this is an hypothetical case, and the endpoints and the groups and the design in general of the study, it's up to the applicants. What I think we have to have in mind is that we should try to demonstrate the safety and efficacy, and this is our objective, and this is the thing that we need to have in mind when we design the protocols, and of course have in mind the 3 Rs, and of course we are here as scientific advice groups, and we are here for support you during development. Thank you.
Then we have a very long question. I would like to ask about how you mitigate a specific safety risk related to phage therapy in this context, which involves virulence that is mediated by toxins that are often borne by prophages that can be expected to stochastically be transcriptionally mobilized by lytic phage stress. Indeed, most antibiotics are contraindicated for E. coli 0157H7 infections in humans in most contexts, as they mobilize the expression of Shiga toxin borne by prophages, causing more harm than good. How do you ensure that the treated weaning piglets will not be harmed by toxins that could be transcriptionally mobilized from target bacteria by phage stress, regardless of whether the phage is microbiologically effective, given that any effect could be expected to be specific to the interaction of your product with a specific bacteria present in the weaning piglet? Please let me know if I have to repeat something.
Well, I will try to go step by step. So the first thing is that, as we discussed along the day, we prefer the use of phages without this kind of genes, and as I think I shared in my example, these fantastic phages have no prophage and so on. And regarding the release of this kind of genes by the bacteria, I will say that for that is the safety studies are done to see if we can expect any unexpected effect as the ones that you are mentioned here. That's why the laboratory safety studies are needed, even when we think that the product is safe, because bacteriophages are considered safe in general, because they are everywhere. However, studies are needed in order to prevent this kind of situations that you are describing in your question. The design of the study should try to be based on that risk that we can forecast, but of course there are others that we cannot, and that's why they are needed, and that's why they are asked by regulators. Thank you.
Thank you for that. I think a hand was raised. We're not totally sure whose hand it is. So the name seems it's you.
Hello, Frédéric Vieville from Ferricides Pharma. So thank you for the today's discussion and this open-mind regulatory text with a flexible composition. So that is very great. Today, we didn't talk a lot about phagogram, so this in vitro diagnostic test to define and to verify the susceptibility of the bacteria pathogen strain to the proposed phages. So we can see a slight difference between the veterinary guideline, which propose the phages for prophylaxis and metaphylaxis, and the pharmacopoeia text propose the bacteriophages to cure, to treat. So I think there is a quite difference between the two texts, and my question is perhaps is to avoid any phage resistance in human, and that lead to my concern and point. It's regarding veterinary use. It seems that the phagogram is not mandatory afterwards to treat or for prophylaxis in a farm, and this could lead to phage resistance if we spread some bacteriophages, and phage resistance also for humans. So how you address this concern? You see? You understand my question? Yes.
Okay, thank you for this question. It's really interesting. So about phage resistance for humans derived from the use of these bacteriophages in the veterinary field, I will say that it's a bit different. There is a big difference between antibiotics and bacteriophages. However, the risk is there, of course, it is there. And I think we will gain experience when we have, I hope soon, some applications. However, I think the situation is different between antibiotics and bacteriophages, because bacteriophages are very specific and they are auto-limited when they affect the bacteria. It's not the same for antibiotics. Antibiotics remain there and they are not auto-limited. So I think the risks of release of resistance of bacteriophages is not so high like in antibiotics, because bacteriophages are not widespread like antibiotics. So the risk is there, of course. We need to characterize it. The applicants need to characterize it, but we expect that the risk will be lower. Thank you.
Thank you.
May I? In the veterinary field, the antibiogram is not mandatory also. So for a novel therapy or for a product, a new product, it would be difficult to be more stringent than for antibiotic in veterinary.
Thank you.
Yes, thank you, Frederick. I think we've certainly, this issue has certainly been discussed, but on the one hand you want to reduce all risk, on the other hand you don't want to limit development of such products, especially since they may replace antibiotics with which you also have problems. So yeah, there's a narrow line there. There was another question from Dr. Ruby, as I understand. Please go ahead. Dr. Ruby, we cannot hear you, so it could be that...
No, thank you. It was the wrong raised hand. Something is popping up. I don't know why. Sorry.
Okay, that's fine. No problem. Were there any other questions?
Yes, there are still some questions in the chat. So one question was where the phages against the otitis pseudomonas first were isolated from.
From my mind. Sorry, it's just hypothetical example, so I have no response.
Okay, so I go directly to the next question. The alteration of gut microbiota raises the question of phages which are planned to destroy regular enterococci flora for breeding reasons is estimated as being unethical all over and will not be allowed furthermore. Is that so?
I think we all have difficulty understanding the question, but I derived that the question is on whether it would be ethical to treat non-diseased animals to alter their gut flora for other purposes than preventing disease. Well, the only response I can give that in the regulation, veterinary medicinal products can also be veterinary medicinal products if they alter the physiology of an animal, not necessarily associated with the disease. So that is the only answer I can give at this moment. But obviously, for any product, there has to be a positive benefit risk.
And maybe it's up to the veterinarian to make its own balance of benefit and risk about such a product and to administer this preventive or treatment or not.
Then another question related to the gut microflora. The gut microflora composition is likely not suited as a measure of the safety of phage products. Gut microflora is constantly changing due to feeding regimen, feed composition, animal handling, and stress. Relevant physiological or clinical effects of the phage therapy product should rather be used, e.g. diarrhea.
Thank you for the comment. I will say that in the study was a control and treated group are proposed to be used in order to compare between these two groups. Then you can, by this design, to pass through the modifications of the flora derived by other factors that are not the bacteriophages. Thank you.
Thank you. Then there's a comment. Efficacy and safety endpoints have to be very well established / planned when preparing a study protocol. And then another question. In the otitis externa field study example, the control group received saline and not a VMP authorized for the treatment of otitis externa in dogs. Is this really possible?
This is possible. It's preferable, maybe for ethical reason, to have a comparator, but for this kind of treatment, it could be maybe difficult to show that the treatment is as efficacious as the comparator.
Yes, I think in general, if well justified that a negative control group would be preferred, then it is possible. But indeed, generally positive control groups, if available, are used. You have more further questions?
Yes, there are further questions. One question for the EDQM representative. As a lawyer, I'd like to stress the human rights issue on getting easy and affordable access to life-saving cures. If a maximum safety level pushes costs to astronomous heights, would it then be justified to switch to a reasonable level of safety in order to get the cure at all?
If I may comment, it's a very interesting question, but this is a veterinary forum, I think, and perhaps if questions on human rights to the EDQM are asked, perhaps they can be given in writing to the EDQM as a comment to the guideline. So I think with that we have reached the end of the list of questions, or is there
yeah, there's one other question which is not
the last one.
Yeah, which okay. If indication is authorized for monophage treatment, sort trials, can other phages from the flexible composition be applied sequentially one after another on the farm?
I think we've answered this sort of question before from a question in the room, and the conclusion was yes. When you do this in clinical trials, you have to take such things into account into the design beforehand. So not change the clinical trial as you go, because that would decrease the value of your clinical trial. So in principle, it could be done. So with that, I think we have reached the end of the journey here. Again, I would like to thank all of the participants and specifically all of the speakers. Thank you very, very much. Well done. I think we've had very good discussions here today. I will not summarize everything, but I will just leave you by stressing that please comment to the guideline in writing. I'm sure you will find how to do it because you've also found this meeting and entered it. So even if you now realized after these discussions that we mean a certain thing, but you initially didn't understand that from the guideline, perhaps even then you can comment on that so that we will be able to clarify the text if this is necessary. So that would also be helpful, so that upon first reading, it will be clear what is meant to everybody. So any comments there or any suggestions are really helpful. And with that, I will leave you. So thank you very much.